Is my screen visible?
No, not yet.
No, not yet.
No, not yet. Okay, just a sec. How about now?
Yeah.
Yes.
Yeah. Yes.
Okay. Okay. All right. So good morning, everyone, and thank you, Dr. Kishore, for the kind introduction. So yeah. So before I start, let me thank the organizer for this international colloquium, Dinhata College, to consider me as one of the speaker, and I'm really honored and flattered to be here. So what I am going to talk about today is basically my journey during my PhD, where I work on engineering antimicrobial surfaces to combat diseases. So basically, I have outlined my talk here. So first, I'll just very briefly talk about infectious diseases that are caused by different pathogens, and then we'll ask some of the basic questions, like how do the infectious diseases spread or occur? And then how can we combat these infectious diseases? And more importantly, how can we prevent or reduce the spread of infectious diseases? And then finally, I'll talk about some of my PhD work that I worked on chemical engineering of surfaces in combating these infections. And in the end, then I'll conclude my talk. So here, just to give you a brief introductions, I think we are all familiar about the diseases, the infectious diseases that are caused by the either residential or foreign invaders. And invaders, I mean to say, is basically bacteria, fungi, or viruses. So here I've just shown some of the examples that, the diseases that are caused by different types of bacteria, like bacterial meningitis or respiratory tract infection, or even urinary tract infections. Here are some examples that are caused by some infections that are caused by fungi. Here, for example, the skin infections, or even the infection in the nail, or the viral infections. I think we are all familiar about the common cold and flu virus, which is caused by the influenza here. And this I've shown along with the other different signs. And this particular virus needs no introductions, as we are all familiar about the coronaviruses, which causes COVID-19 diseases. So we are already in the middle of a pandemic, and how serious and a global health issue it could be. And this is an example that we can look forward to, or we can think about it. So, but if we just look at this old map here, so technically infectious disease has always been, in fact, a global health crisis, a global health issue. So this chart here shows the different types of infectious diseases, for example, the tuberculosis, HIV, diarrhea, malaria, dengue, and all others. And the different countries have suffered different types of diseases, and it has always been a serious health concern, a public health concern. Now, more importantly, if we just look at this chart here, this clearly tells us that how infectious disease can be a serious concern, or is actually a serious concern. This shows that out of every 100 deaths, 20% or 20 deaths is basically due to the infectious disease. So almost 20%, which is double than the death caused by the cancer, or even it is almost equal to the cardiovascular diseases, and higher than any other cause of death. So this tells us that infectious disease is basically a very serious public health concern. So coming to the next aspect of my talk, so how do the infectious diseases actually spread or occur? So this is very important to know, because if you want to really combat the infectious disease, one needs to know if we can prevent these diseases rather than going for a treatment. So here I've shown some of the important or basic modes or ways by which infectious disease actually spread. So some of the basics are like air mediated, surface mediated, it could be water or contaminated food mediated, it could be vector mediated, or it could be the direct transfer. Now the air mediated, as obviously this picture shows here, that the virus, bacteria, or fungi that could be sprayed out from us, like when during talking, sneezing, breathing, or even coughing. So these aerosolized particles can come out and then it can leave and stay in the air or even get deposited into the nearby surface. And it can go as high as 16 feet, but technically it travels around 2 to 4 or even 5 feet. So this basically makes sense that when we talk about like social distancing due to coronaviruses, at least a minimum of 6 feet distance is required, because these droplets can travel up to 6 feet, which can cause a direct transfer from one infected person to another non-infected person. Other than that, this direct air mediated, these droplets can also get deposited onto the surface. And surface, I mean to say any object that is surrounding us, for example, table, chair, floors, walls, computers, keyboards, anything that is near, surrounding us. And then they can stay there for, like hours to days to even months. They can survive there. And once a person touches this area, and then in turn, if he or she touches his nose, face, or eyes, and then this infection can directly be transmitted to that person, actually via surfaces. And then the other ways, like if there is contaminated water or contaminated food, that can also lead to a transfer of infection from one person to the other via this water or food. Then there could be a vector mediated. I think we are all familiar, this vector mediated mosquito, which causes dengue or even malaria. So it basically carries from one person and delivers to another person. So that's how they act as a mediator or as a way of transferring infection. And then there could be a direct transfer if the blood, let's say, for example, during the blood transfusion, and if this blood has been collected from a person who was already infected, and then now it's been transferred to another person. So the person who is receiving it, he can directly get this infection due to this contaminated blood transfer. So among all these modes of spreading infections, if we just summarize everything, and if we look at this stat here, this chart, which says that 75%, so 75 to in fact 80% of these infections actually spread through the contaminated surfaces. So this tells us that how the surfaces are so important and in fact plays a vital role in transferring or transmitting infection from one person to the other. So ideally speaking, if you want to combat or if you want to prevent the infection, so if we make the surfaces antimicrobial, then technically we should be able to prevent the infection. So that was the whole idea here. And when we talk about the surfaces, we can further subcategorize into different types of surfaces. For example, when we're talking about surfaces of non-living objects, for example, the rooms, walls, floors, etc. So this is like, we call it as abiotic surfaces. And then there is other type of surfaces, like for example, the implanted devices or like other implants which are being implanted into the human body. Let's say, for example, the artificial heart valve or pacemaker, or let's say titanium implant to hold the fractious bone together. So these implants also can directly cause the infection, because if they are contaminated, they can act as a transmission of infection. So these are all abiotic surfaces. Then there could be abiotic surfaces, like for example, the skin infections. This is from like a living or human body. So it's basically we call it abiotic surface associated infections. And here the skin infection is one such example. And also this is a superficial example, a superficial infection. So this is one type. The other type that could be a deep tissue or internal organ infection. And in fact, when we have multiple organ infections, it can lead to sepsis. And that is, in fact, the multiple organ infections are actually called sepsis, which eventually leads to the death. It's very difficult to, in fact, treat the multiple organ infection. So this deep tissue infection is, in fact, a very serious issue. So what I'm going to talk about today is I will give you one example for each class of surfaces here that I talked about, these 4 different types of surfaces. So I'll just give one example, how we can develop different types of material and, in fact, combat the infections that are associated with these type of surfaces. So the question is then, how can we combat the infectious diseases? So of course, like when we have a bacterial infections, we can go for an antibiotics, but even not, like all the antibiotics are not even, the antibiotics are not even active against all types of bacteria. So we have very limited resources of antimicrobial agents. And more importantly, the antibiotics are not active against virus. And most of the antibiotics, are very less effective against fungal infection. So we need something which is very broadly antimicrobial, which basically kills virus, bacteria, fungi, everything all together. So we call them as a broad spectrum antimicrobial agents. So technically, how do we design these broad spectrum antimicrobial agents? And in fact, we draw inspiration from nature in our body. For example, the eyes. So our eyes is the most exposed organ, but still we do not often get the eyes infection. So we wonder like why and how is this happening? So it's basically we have lots of antimicrobial peptides in our body, which acts as a defense mechanism in our body. So basically, if we look at the structure of these antimicrobial peptides, they have three basic components. They have one is the positive charge, the number of positive charges that generally varies from 2 to 9. And then they have hydrophobic domain or hydrophobic moiety. And there is a fine balance between the positive charge and hydrophobic moiety, which is amphiphilic balance. Now, how can they be like active against a broad spectrum of antimicrobial for microbial pathogens? So for example, if we look at the structures of bacteria or even virus or fungi, so basically they have a lipid membrane, and this lipid membrane is basically nothing but a phospholipid bilayer like this. And further observations, if we look at, so this phospholipid bilayer, so they have this hydrophilic head and then this lipid tail. So here, in most of the cases, like for most of the pathogens, this hydrophilic head is actually negatively charged because of this phosphate group, because the end functional groups are basically neutral, so which makes the head group overall negatively charged because of the presence of this phosphate group. But on the other hand, in the case of mammalian cell, this is mostly deuterionic. So one negative charge from phosphate and one positive charge from these, like this positive ions or ammonium ions. So basically overall, so the majority of the phospholipids in mammalian cells are actually deuterionic. On the contrary, majority of the phospholipids in pathogens are actually negatively charged. So what actually happens, these positively charged antimicrobial peptides, they have a fine electrostatic interactions with the negatively charged lipid membrane of the pathogen, and as well as they can interact with their hydrophobic domain through van der Waals interactions with the hydrophobic bilayer of the lipid membrane, thus causing force and literally destroying or damaging the pathogen membranes, and therefore killing a very broad spectrum of pathogens because they all have membranes, and so they're very broad spectrum. So, but technically or practically, to produce this type of antimicrobials in laboratory or to use clinical settings, it's very critically challenging and as well as very costly. So ideally, we wanted to create mimics of these antimicrobial peptides and use these synthetic mimics to tackle this variety of microorganisms, thus developing a synthetic mimics of broad spectrum antimicrobial agents. And again, we follow their structural or functional similarity. So basically, again, in our design, what we are going to have is basically positive charge, hydrophobic moiety, and there will be a fine balance between hydrophobic and hydrophilic moiety. So that's our design criteria to develop a broad spectrum antimicrobial agent, keeping in mind that most of the pathogens membranes are negatively charged. So now moving on. So the next questions that we're asking, so how can we prevent or reduce the spread of infection? So as I talked earlier, that most of the infections actually occurs through contaminated surfaces. So if we can make the surface antimicrobial, then we should be able to prevent or the spreading of infections. So yes, of course, the idea is like basically we have an antimicrobial surface and we kill the pathogen there, so we prevent the infections. And of course, the prevention is better than the cure. So technically now the questions that we're asking, so how can we engineer the surface to combat the infections? So there are different actually strategies that are.
Hello, Jia. You are muted. Please unmute yourself.
Jaul, you'll have to just unmute your computer.
the surface is antimicrobial. So here, there are different strategies already there to create antimicrobial surfaces. So for example, in this first strategy, what is done is we take different types of antimicrobial agents and then just simply paint or coat onto any surfaces. So what happens is over time, these antimicrobial agents leach out or release out from the surface and kill the microorganisms in the surroundings. However, there is a problem to this technique. The one is there is always a release, unwanted release, whether the surface is contaminated or not. And at some point of time, all the antimicrobial agents are going to be released out, and eventually the surface will be left with no more antimicrobial agents, and therefore this strategy is non-permanent. So to overcome these problems, another strategy was introduced. So it's called a covalent coating of antimicrobial agents. So we do some sort of chemical reaction onto the surface itself to immobilize different types of covalently onto the surface. So they are not leaching out, they're not coming out, they're there. And when there is a bacterial or any other microbial contamination onto the surface, the antimicrobial agent kills the bacteria. However, this technique has its own limitations. For example, it requires multiple synthetic steps to do surface modifications. And then different synthetic strategies is required. For example, if we have a wooden surface, if we have a steel surface, if we have other metal surface, or maybe a cement surface. So different surfaces requires different types of chemical strategies to do the surface modifications, which practically makes it very impossible, in fact, or very challenging to do so. And then also it requires a very trained personnel, either a chemist or a trained personnel, to do these sort of modifications. So this strategy, even though quite effective, but practically it's quite challenging. So we wanted to come up something with a different sort of strategy where we wanted to have a permanent coating, but at the same time, the coating is very simple, just like paint the surfaces. So we came up with the idea of developing a water insoluble and organosoluble, means these antimicrobial agents are soluble in organic solvent, and at the same time, they are insoluble in water, so that we can just simply paint these or coat these antimicrobial agents onto the surface, so they are not going to come out as because they are water insoluble, and at the same time we can have a permanent antimicrobial coating with a very easy coating procedure. So that was the whole idea, and that's what we call it as a non-covalent coating of non-leaching antimicrobials. So here there are obviously it would offer the advantages like a single-step coating, no leaching, and no need for a trained personnel. In fact, anybody can just simply paint anywhere onto the surfaces. And again, so we kept in mind, as I discussed before, that there are several design criteria that we need to think about it when we are designing these antimicrobial agents. So of course, we need to have positive charge, we need to have hydrophobic moieties, and there has to be an optimum balance between hydrophobicity and hydrophilicity. And now the fourth criteria that we are introducing is, at the same time, these antimicrobial agents or polymers should be water insoluble and soluble in organic solvent. So here is one of the strategies or reaction methods that we developed in our laboratory, and we came up with a water insoluble and organosoluble polymers. So we took a commercially available polyethyleneamine and did some chemical reactions to introduce positive charge, to introduce this sort of long hydrophobic alkyl chain here. And then we also varied the degree of quaternization, the chain length by which we were able to make both water insoluble and water soluble polymers. Like for example, if the chain lengths are like this, we get a water soluble polymer. If the chain length are like this, then we get water insoluble polymer. We also varied the molecular architecture. I mean to say that branched polymer, linear polymer, and then different molecular weights we used. So a variety of polymers were synthesized. And then finally, we tested their antimicrobial activity with the water insoluble ones. So how do we test it? So to mimic the natural scenario of bacterial spreading and deposition, what we did is we took the glass surface like this, and then we just simply coated these glass surfaces from the organic solutions of our polymer, like here. And then to mimic the spreading of microbial species. So we took the bacterial suspension in this chromatographic sprayer, and then we spray the bacteria both on uncoated and our polymer coated surface. You see, this is the results after 24 to 48 hours. So each white dot basically represents one bacterial colony. So this clearly indicates that the plain surface is not antibacterial. On the other hand, our polymer coated surface under the same experimental condition showed even zero colony, showed no colony here. So technically it showed its lethality against the bacteria. And not only bacteria, we tested several different types of bacteria along with fungus, a different variety of fungus, and also we tested against the virus, influenza virus, and we found that this particular polymer is lethal to all types of pathogens that we tested. And one of the polymers were found to be the best here. So not only that, this was just a direct coating of our polymer onto the surface, but we wanted to have a real application. So what we did is we took the commercially available paint, and so this is like Asian paint we took, and we introduced a certain amount of polymer with the paint, and then we coated the paint onto the glass slide, and the same way we sprayed the bacteria, and we wanted to see how does it work. So here, this is the plain paint coated glass surface, and obviously we see a lot of dots here, which indicates the growth of the bacteria. On the other hand, paint plus our polymer coated surface showed no bacterial growth on the surface, the kind of validating our ideas of having a permanent.
Hello, hello, Gio. Hello, again it's muted. Could you unmute again?
Idea why this is happening. Can you hear me now?
Yeah, that's okay.
Okay. So was I muted like too long back, or just now it happened?
Just now.
Okay. Yeah, okay. I don't know why this is happening. I'm sorry. Yeah. So okay. So yeah, so I was talking about that how these polymers actually is responsible for killing the pathogen. So here we first wanted to see what happens onto the surface. So here this is how this is the E. coli bacteria, the rod-shaped E. coli bacteria. This is how they are on the plain surface. But if we look at on polymer coated surface, the bacteria, there are holes and pores, and the structural integrity is lost. So technically, the bacteria is totally destroyed here due to the presence of our polymer. And then we also further tested that whether their membrane is compromised or not. So we selected two dyes. One is the green dye and the other is the red dye. So the green dye, what it does, it can permeate the live bacterial cell membrane, and therefore it can stain the live bacteria with a green fluorescence. On the other hand, the red dye can only go to the cells where the membrane is compromised. So it can only signify the dead cell or show the dead cell. So basically, this is our plain surface, and the presence of green signal clearly indicates that most of the bacteria are alive here. On the other hand, on our polymer-coated surface, these bacterial membranes are compromised due to the presence of our polymer, and now they're giving the red signal. So it's basically further validates our concept that these sort of polymers can positively charged, hydrophobically modified polymers can interact with the negatively charged bacterial cell membrane and thereby disintegrating or compromising the membrane integrity. So having said that, now I'll come to the conclusion of this particular section here. So basically here we developed a water-insoluble and organosoluble cationic polymers with a new synthetic methodology, and we used it as an antimicrobial coatings, and we believe that this sort of paint or this sort of polymers can be used to apply on floors and then all operating rooms, hospital equipments, etc., everywhere to make the surfaces antimicrobial. Now coming back to the next question here, the next aspect. Now, for the implantable devices which are being used into the human body, we can't use something which is like non-degradable and which is like very toxic. So we need to have an antimicrobial polymer which are biodegradable. Do work, they degraded in the body, and also they are non-toxic. So for these purposes, we chose a naturally occurring polymer known as chitin or chitosan, and they are available from this sort of shrimp or crab's exoskeleton. So, and human enzyme lysozyme is actually known to degrade this particular polymer. So here, and also this polymer is known already for different medical or clinical applications, like for example, as an artificial skin, wound dressings, or even in contact lenses. However, the problem with this polymer is that this is very insoluble in all the commonly used organic solvents, so it cannot be used just directly to coat onto the surface, and at the same time, its antimicrobial activity is very poor. So ideally, this is not like a suitable polymer. So we wanted to use this polymer and then develop a highly water insoluble and organosoluble polymer, and at the same time we wanted to increase its antimicrobial activity while keeping its biodegradation and other properties more or less intact. So this is how we do the chemical synthesis. So I'm not again going to the details of the chemical synthesis. We do some 2 to 3 steps of just simple chemical modification to introduce the positive charge and as well as long hydrophobic alkyne chain, so that it can have interaction with the bacterial cell membrane and at the same time make this polymer water insoluble. So having synthesized different types of polymer, and then we tested their antimicrobial activity in the same way as we tested before. So here, as again, like the previous polymer, we see our modified chitin polymer also showed very high antimicrobial activity against a variety of species, the pathogen species that we have tested. Then we wanted to test whether this, due to the chemical modification, are these polymers still biodegradable or not. But when we throw them into the lysozyme solution, we see these polymers are degrading over time, and 10 to 15% degradation we observe probably in 15 to 20 days. But this is in vitro conditions. We wanted to test under in vivo condition what happens. So we made a polymer disc like this and put it into the rat's body subcutaneously. So this is how they look like under scanning electron microscopy images. So this is at the beginning, and then this is from the body harvested after 14 days. So this is how they look, and after 21 days and after 28 days. So what we are seeing here is basically that with time, the surface is becoming non-homogeneous. There are holes and pores into the surface, so clearly indicating that our polymer disc is degrading over time. So basically, even after the chemical modifications, we are able to retain their biodegradation property, at least to some extent. Next, we wanted to evaluate these polymers' activity under in vivo condition. And to evaluate the real application, what we did is we took a medical device, for example, the polyurethane catheter, which is used as urinary catheter in the body. So we took this polyurethane catheter, then we coated with our polymer, and then to mimic the natural scenario again, so we incubated this coated catheter with a bacterial suspension to contaminate this catheter, and then finally we implanted it into the mice body, and after 4 days we take out this catheter, and then we look the catheter surface, and also we did the bacterial counting as well. So this is how the catheter surface looked like. If we just expand this area and see it here, we see this is like a huge cluster of bacteria on the non-coated polyurethane catheter. But when we coated with our polymer and then infected with the bacterial solution, and after implantation and 4 days of harvesting, then 4 days later when we harvested the catheter, we see no bacterial colony or no bacterial cluster is present on our polymer coated catheter surface. We also count the bacteria here, and this clearly shows that with increasing the polymer coating, the bacterial count dramatically reduces, which further indicates that our polymer is able to prevent the microbial or bacterial contamination when they are coated onto the medical device, such as catheter. This is a further histopathological analysis, which I'm not going, again, it's just basically indicative of this, supportive of the other two results I just explained. So moving on, so if to summarize this particular project. So we again developed a water insoluble, organosoluble, but a biodegradable cationic polymers. And we believe that these sort of polymers potentially can be used in different biomedical devices to prevent the biomaterial associated infections. And so we find this work, we patented this work in several countries, and at the same time, this is the work that received the Gandhi and Young Technological Innovation Award from Government of India. And in fact, now this work has been taken further by Department of Science and Technology, and they wanted to develop by using our polymer now to have an antimicrobial cloth. Literally, we can coat it on face mask, we can coat it in different types of mask, or even the everyday clothes that we use. And in fact, in our laboratory now, there we're trying to do some sort of coating technology by which we can have a very stable permanent coating on any clothes so that we can make the clothes permanently antimicrobial. So moving on, now we just talked about this and this implantable device aspect. Now let's talk about the biotic surface associated infection. Here, for example, the skin infection. So if we just look at some of the antimicrobial gels that are generally used for topical infection, or you would call the skin or superficial infection. So here, this is one antimicrobial agent, which is fuchsic acid-based ointment or gel is used. Then there is a colistin or lipopeptide-based, and there is silver-based gel. And you have the gentamicin, which is a common antibiotic that we always use. So this gentamicin-based ointment, these are all used to prevent or basically to cure the biotic surface, or basically the skin-associated infection. But however, there are major problems with all of these agents. First of all, there is a bacterial resistant development. So the pathogens are becoming more and more resistant to these agents. So most of the time, these agents do not work against the infection. And also, they are not broadly antimicrobial. So we wanted to come up with one single antimicrobial agent which could be used in the ointment, and to make it a universal or very broad spectrum antimicrobial ointment. And to do so, we used, again, the similar strategy of antimicrobial peptide, but this time we developed a small molecular based system, and these small molecular systems are water soluble so that we can make an ointment or gel out of it, along with some other. So here, there are, again, the similar design principle were considered, keeping in mind that we needed an electrostatic interaction, we needed a hydrophobic interactions, and in addition to that, we needed to have an there is a balance between hydrophilic and hydrophobic content or moieties. So here, we had two positive charges in our design, and then we introduced, in the same time, we wanted to make it a biodegradable. So we introduced a non-peptidic amide bond, which should be degradable in the presence of various proteases enzymes that we have in our body. And then we varied the structures in different ways so that we have design 1, design 2, and design 3. I'm not going to the details of this design, but technically we had different types of design just by interplaying the hydrophobicity and hydrophilicity. So here, this is their synthetic methods, and a total of 74 molecules were synthesized. And one of the molecules were in fact found to be active against all these organisms, microorganisms, and with a very minimal toxicity against mammalian cells. So this is an HC50 value, which is called hemolytic concentration, 50% hemolytic concentration. Sorry, this is a hemolytic concentration. So basically, at which 50% RBC lysed the concentration of our compound. So this is like a very high as compared to the MIC, which is called minimum inhibitory concentration of our compound. So this is the minimum concentration that is required to inhibit the growth of microorganism or bacteria. So, and also we showed that our antimicrobial agents with repeated exposure at the sub-MIC level do not allow the bacteria to develop resistance. On the other hand, the commonly used antibiotic like norfloxacin or colistin, so they were shown to develop resistance very fast. In fact, after just few passages. But even after 20 passages of our compound, still the bacteria were not able to develop resistance. So possibly again, because they target the bacterial cell membrane, and it's almost impossible for the bacteria to have a drastic change in their cell membrane composition. That's what we believe, that why the bacteria were unable to develop resistance against these antimicrobial agents. So next, we also tested their in vivo toxicity in different animals. So we tested against rat, rabbits, guinea pig, and then we found the LD50 value, which is the lethal dose, 50% lethal dose. So so it's basically at which like the 50% animal dies or basically suffers severe consequences. So here we have a very high dose, and still we do not see any significant skin toxicity. So thus possibly these molecules are very less less toxic, I would say, or causes less skin inflammation or irritation. Next, we we tested the skin infection model. So what we did is we take mice and then we save the hair, and then we infect with the MRSA, which is methicillin-resistant Staphylococcus aureus, which causes skin infection very often, in fact. And then after 24 hours of mice skin infection, then we started giving treatment with our compound. And at day 5, we took out the skin and looked under scanning electron microscopy, and at the same time, we determined the bacterial count. So initially, the bacterial dose was given around 10^8, and then after 4 days, what we see in the case of untreated one, there is a huge increase of bacterial count, so it's like almost 10^10. But in the case where we treated with our compound, this is like almost a detectable level. This is the lowest detectable level that the method that we use. So it's almost eradicated the infection on the mice skin. But when we use some of the commercially available antimicrobial gel or ointment, it shows a very minimal effect. And this is the ACM image again, as clearly it is shown here that there is a huge cluster of bacteria. Hello? I can't hear. Am I audible?
Yes, yes, you are. You're
Okay.
audible.
Okay, thanks. Okay, so moving on. So in the case of treated skin, we see very minimal amount of bacteria here. So again, this clearly indicates that it has almost eradicated the bacterial infection. So in fact, we took our compound, this one, and we made a ointment, and that results I have not shown here, but basically this sort of cationic small molecules can be used to develop different types of ointment and can be used as a safe and non-toxic antimicrobial agents of ointment formulation. So coming to the final section of my talk here. So this, when we have deep tissue infections, like as I was talking about the internal organ, in the case of open heart surgery, we can have infection in the organ, and we can even have in the deep tissue, because this surgical procedure is very vigorous and extreme. So ideally, so what it does, so what we do generally, so after this infection, so the doctor do the stitches like this, and then in most of the cases, they apply a commercial sealant. So this is the only clinically approved and available sealant. So this sealant, what it does is basically, this is like an injectable solution, but the moment it is injected, it forms gel here, and it basically fills the empty spaces here. That's why it's called sealant, because it helps to seal it. And at the same time, it also partially acts as a bioadhesive, because it can interact with the surrounding tissue and hold the tissues together, and that's why it acts as a glue, which is called as bioadhesive. And the other advantage of this one is it can also stop the bleeding, and that's why it is hemostatic. However, this commercial sealant has some limitations because it is not inherently antimicrobial. So doctors suggest lots of antibiotics as a precautionary measure to prevent any possible infection into this injured or surgical site. And it's also like this sealant has been shown not to promote wound healing, but this is a serious wound, and we need to heal this wound as fast as possible as a recovery measure. So ideally, we wanted to come up with an alternative commercial sealant, which has all these 4 properties in one system. We call it as all-in-one. So our design principle again, so we developed a water-soluble antimicrobial polymer by introducing a positive charge in the polymer, and then we have a water-soluble adhesive polymer which has some functional groups, and these functional groups can react with the amine groups of the tissue's protein and thereby act as an adhesive polymer. And at the same time, we have some free amine group in our antimicrobial polymer, so they can also react with this polymer, leading to a formation of gel the moment we inject through a dual barrel syringe. So this is a dual barrel syringe. So in one syringe, we have one polymer solution, in another syringe, we have another polymer solution. And when we inject them together with a single syringe at the end, so that the moment they get mixed, they form a gel. And this is the example. As soon as we mix them together, they form a gel. And these are some of the hydrogel characteristics where I've shown here, which I'm not going to the details of it. So next, we wanted to test this polymer, these hydrogels, do they act as a hemostatic material. That means, can they stop the bleeding? And one of the most commonly used method is the liver puncture method. So what we did is one of the lobes of the mice were punctured, and this was as a control one. So we measured the amount of the blood that is coming out after puncturing the liver's lobe here. So this is the amount of the blood that comes out, and this also evident from this image. But after puncturing, the moment we applied our material, the bleeding was immediately stopped. So we also determined that how much, like the blood came out. So it's basically, it shows that there is a significant amount of less blood loss. That means this polymer is acting as a potent hemostatic material, or stops bleeding immediately after the injury. The next, we wanted to see how strongly this polymer, or this hydrogel in fact, hold the tissues together. So we take 2 pigs' skin and then apply our material in this common area, which is this colored common area, and then we started pulling these 2 skins apart and measured the force by an instrument called dynamic mechanical analyzer, and it gives us the force that is required to detach these 2 skin together. So here, this is the force which we get around 5 to 10 kilopascal, which is much higher than the commercially available sealant, is this 3-4 kilopascal approximately. So we have hemostatic ability, we have adhesive property. Now we wanted to see whether we can prevent the internal organ infection or not. So one of the common way to do is called Cecum, ligation, and puncture model. So Cecum is a small part of the intestine, which is in between the large and small intestine, and it contains just after 24 hours. So the bacteria has come out and has infected that area. It has become totally dark and black. And also, in the case of treated cecum, you see it actually shows its normal color of the intestine. So this clearly tells that this is able to at least prevent the leakage of bacteria and also prevent the initial infection. Now we wanted to see, can we really prevent the sepsis? So that we did following the 8 days of this Cecum ligation model. And we had several groups of animals, and in each group we had 8 animals or 8 mice. So in one of the groups, which is just the Cecum ligation, but no material is applied, after 8 days, only 1 animal survived among 8 animals. But when we applied our gel, we see 75% survival. That means out of 8 animals, 6 animals survived there. So this clearly indicates that this material is able to prevent the sepsis in the under in vivo condition. So next we wanted to see, can this material promote wound healing? So here what we did is, this is done in rat. So we removed around 16 to 18 millimeter of the rat skin. This is called skin incision wound healing model. And then we applied our material, and in the control we do not apply any material, just the saline as a control was used. So here, in the case of our gel, so this is how we're looking at the wound site with time. So at day 5, day 10, day 15, and day 20, we see at day 15 to day 20, the wound was almost healed as compared to the control or even the commercially available sealant here. So this again says that this material promotes the wound healing of the infected, of the wounded area. And this was the quantitative measure of how much wound is being closed at different time points. So as obvious, over 80% wound was closed just within 20 days, as compared to just around 50 to 60% in the case of control. So it's basically promotes wound healing finally and increases the rate of healing process itself. So finally, then I come to the conclusion of this section is that we have an intrinsically antimicrobial, hemostatic, and wound healing material, and we believe this could be used for surgical site infection and even in the military conflicted area where we need to stop the bleeding immediately. So that's what we believe for this material. We find the patent application for this, and now we're talking to the DRDO to get it a military project, so where it can be used any sort of formulations for the prevention of any infection or promoting the wound healing when we have a deep cut or injury. So finally, I come to this, so I acknowledge my PhD supervisor and my current postdoc supervisor and other people I have not shown here any of my postdoc work, but I'm working on musculoskeletal disease model. I'm trying to develop different types of material and trying to see whether we can do something to treat osteoporosis and osteoarthritis. Anyway, maybe some other time I would like to talk about this. So I'd like to also acknowledge the different funding agencies. And this is my building somewhere. I work here, and this is my current lab. And I would personally like to acknowledge and really my sincere like my wholehearted thanks and like gratitude. I would like to convey my gratitude to Dr. Kishore Thapa, who personally approached me, and my brother, who is already a member of the Dinhata College, and my friend Dr. Bappa Sarkar. We are not only the same batchmate, we're like very close friends, and we did our masters together. So I know him for a long time, and it is really nice to be here. And I once again thank you all for your kind attention, and I will be happy to take any questions if you have.
Okay, thank you, Yaul, for your very eloquent talk, and in a very simple way you have mentioned all about this effect of antibacterial and antiviral surfaces in the prevention and spread of infectious diseases. And just before I invite Dr. Santos, I would just like to request Dr. Santos just to bear with us for a few minutes, because there are so many questions, but I would like to select one or two questions only due to the shortage of time. The first question I want to put to you is that Dr. Gauri Das, she writes that in hemostatic study, when you use mice for liver puncture, what happens to the mice?
Oh, they are alive. So they are in the anesthetized condition. So breathing, and it was done when the animals were live. And after the experiment was completed, then we sacked the animal. Am I audible?
Yes, you are audible.
Yeah. Okay. So that we wanted to do it when the animals are like alive. So basically, we wanted to see what happens, because so they should be breathing, they should not be dead. Otherwise, that experiment can lead to a default or maybe a different result. So it was performed under anesthesia. Well, it was performed like under anesthetic condition, and they were very much alive when we performed the experiment, and they don't die immediately when we puncture their liver.
Okay, thank you. Another question is from Anuradha Sinha. Her question is, excessive hand washing is causing problem to very sensitive skin in this COVID-19 crisis. How do you deal with it?
Oh, exactly. That is a very good question, and in fact, it's a serious issue because the antimicrobial agents and some of the other chemicals that are used as ingredients in those antimicrobial hand washing or formulations, those are the ones that's causing actually the irritation. So basically, we need to have, like, in fact, we were trying to come up with an alternative antimicrobial agents, which you already have, and now we need to have a like kind of formulations which does not create any sort of inflammation. So we're trying to use like aloe vera gel, and then use our antimicrobial formulation, which we have already tested in different animals that do not show any skin irritation, skin inflammation. So there are different standard models that we have used. So hopefully we should be able to do it. But yes, that is a serious issue, and it can cause inflammation to many people, particularly with a sensitive skin.
Okay, thank you. Another question is from Sunil Sapkota. Are these polymers economically feasible to in access of the general public?
Yes, it is. In fact, that is one of the reasons we wanted to use a commercially available polymer. These are very cheap polymers, and we are doing only 1 or 2 step of chemical modification. So in the end, we in fact evaluated the cost of these polymers, and these are still way more cost effective. I mean to say it's cheaper. So it is practically very much feasible to use. And this is one of the reasons, in fact, we have collaborated in India, I think in Bangalore, one of the pharmaceutical companies, and they are developing these polymers, and in fact they wanted to test for real applications. So hopefully something would come out. So one of the antimicrobial paint polymer and the other one, the last hydrogel that I showed, these two were taken by a pharmaceutical company, and they're now evaluating the possibility of practical applications.
Hello, Kishore sir. Please unmute yourself. Hello, Kishore sir.
Hello. Yeah. Okay. Am I audible?
Yeah.
Okay. Due to the shortage of time, and we are running behind the schedule, so I would like to put all the questions that I have with me to Dr. Jiaul on his email, and maybe he will be able to answer you personally all those questions. Okay.
Sure.
On behalf of the organizers, I thank Dr. Jiaul Haq for his gripping and eloquent presentation on such an interesting topic.
Thank you
He
so much.
highlighted different strategies for creation of permanent antibacterial and antiviral surfaces for prevention of the spread of infectious diseases. Your talk was indeed very relevant in the current scenario. Thank you once again. Thank you, Jiaul.
Thank you so much. Yeah, thanks.
Our next speaker is Dr. Santosh. For that, just as I have already mentioned, that Dr. Santosh is from Birbal Sahni Institute of Paleosciences, Lucknow, which is a very premier institute, and I hope it is the only institute specialized or specified into one particular subject or one particular discipline, that is the paleoscience. And initially, it was known as Birbal Sahni Institute of Paleobotany. And I just like to invite Dr. Santosh here on the forum. Just before that, I would like to ask Dr. Bappa Sarkar to introduce Dr. Santosh Saha. Dr. Bappa Sarkar, please.
Yeah. It is my honor to introduce our next speaker, Dr. Santosh Kumar Shah, scientist D, Birbal Sahni Institute of Paleoscience, India. He did his BSc in Botany honors from Kalimpong College and MSc from North Bengal University. He was awarded PhD in Botany from Birbal Sahni Institute of Paleoscience, Lucknow, India. His diverse research interests include dendrochronology, reconstruction, and analysis of climate variability and changes over the Common Era, past drought, flood, river flow, reconstruction and glacial fluctuation, climate field reconstruction, proxy system modeling, Holocene paleontology, and modern pollen climate calibration. And also, he used the statistics data analysis in R programming environment. Dr. Shah has over 50 publications, including 45 journals, articles, and reviews, 4 chapters contributed in edited book proceedings, and 3 scientific reports. He has supervised 6 MSc dissertations and 3 PhD theses. He has experience in running a number of in-house and funded research projects at the national and international level. He is also the recipient of number of awards and honors, some of which are President, Asian Dendrochronological Association, Sharda Chandra Memorial Gold Medal, and Fellow of East Himalayan Society for Spermatophyte Taxonomy. This is but a very brief introduction of Dr. Shah. With this, I request Dr. Shah to start his deliberation. Please, Dr. Shah. Hello. Hello, Dr. Shah.
Santosh, you're not audible. No.
Now you are audible. Hello, Santosh sir. Please unmute yourself. Hello, Santosh sir. Please unmute yourself.
हेलो सन्तोष आवाज आउँदैछ
Dr. Shah, kindly check your headphones if you have connected them.
हेलो भाइ आवाज आएको छैन तपाईको I think Dr. Santosh is having some problems, so he's logging again. Okay, he's joining sometimes.
Now it's okay?
Yeah, now it's okay.
Okay. Okay. Now is it okay?
Yes, Santosh, it's okay.
Okay, thank you very much. Sorry for some communication gap. So first of all, I think it's visible, no?
Yeah.
Okay, sorry. So first of all, I would like to thank organizer Dr. Kishore Thapa and joint convener of the webinar, Mr. Manjil Gupta and Dr. Bappa Sarkar. And I would also like to thank Chief Patron and Patron of this webinar, that Vice Chancellor Dr. Deep Kumar Mukhopadhyay as a Chief Patron, and Patron Dr. Abdul Kader, Registrar of Dinhata College. So without wasting time, I would like to start my talk. And my talk is on the climate change in Himalayan region, its assessment and future perspective. So for this talk, I would like to talk on the climate and climate change analysis. So because everybody and nowadays the interest in climate change issues, and just now even my 5 years daughter is here to listen about climate change. So you can imagine how the climate change is important. So I know that you have diverse audience like professional. So I want to talk some basic things and also some advanced things. So the basic is what is climate, and before that, you have to understand what is weather and climate. So if you see, like this is a newspaper clipping of like any day. It is reported every day. We see newspaper and it is
Dr.
reported
Santosh,
that.
would you please say your skill presentation in the
No, it's only she's just listening.
no. I screen. Yeah.
Screen is
Say your
okay.
presentation on screen.
It is not visible.
No.
So I think you need to share it again.
Share it again.
I think you need to share your presentation
Yeah,
again, sir.
Where? इधर है तो इधर. Present now. Na?
Yes. Yes, it is.
Is it okay now?
Is it okay?
Yeah.
Oh, it was again. And so the topic of the presentation is climate change in Himalayan region is assessment and future perspective. So if you see what is, first I would like to tell you about what is weather and climate. So as you have diverse audience, so if you see what is weather, then like see in newspaper that a newspaper reported like temperature of the yesterday and precipitation. So all are called weather. Usually the weathers are short-term variation in atmosphere and normally day to day. And suppose if you have weather data for long period, like here I am presenting Lucknow climate data of Lucknow. It is recorded in airport of the Lucknow. So the climate is the average weather at a given point and time of year over a long period. And usually the WMO, that is World Meteorological Organization, reported that classical period is 30 years. So if you have the weather data for at least 30 years, then we call it is a climate or a climate of the station or the climate of the region. And if you see the Earth climate, it is mostly depend between the interaction of 5 different elements or component of the climate system. And these climate the component of the climate systems are atmosphere, hydrosphere, cryosphere, lithosphere, and biosphere. So atmosphere, you see all the gases and that elements, all and hydrosphere like ice, water. Cryosphere is mostly polar ice, that Himalayan ice. Lithosphere, mostly the surfaces, rock surface. These are lithosphere. And biosphere, you know, the vegetation and forest. All this include biosphere. Now, this climate system is continually changing, as we all know, and this is all due to the interaction between the different components. All these 5 components interact with each other, and the climate system work, and also the climate varies or change. But to change the climate system, it should be influenced by some external forcing pattern. Like these are called the causes, like the changes in plate tectonics. We all know the tectonics always affect the climate, but this is the long-term effect. If you see the Earth history, Earth is formed around 4.8 billion years. Okay, and during the entire course of time, plate keep moving. So there is a, due to this plate tectonics, climate changes, but it all occurred in the past. Even the Earth's orbit also changed. And there is a theory or a hypothesis called the Melancholic Hypothesis, which says that there is a, if the Earth orbit changes, there is a climate change. If even if the sphericity also change, even the angle of the Earth also change. So, and it has different cyclicity. Like these changes occur in different time phase, like 11,000 years, 22,000 years, 44,000 years. This orbit and these other things changes. So due to all these things, climate change. And the most important thing is the sun's strength. If you see, the sun is the most important criteria for climate change, because most of the things are controlled by the sun. And when all these forces with all 5 components of the Earth, the changes occur. The changes may occur in ice, changes may occur in water, changes may occur in land, changes may occur in temperature, changes may occur in humidity, changes may occur in pressure. So these all these, due to all these things, climate change. And the most important thing is the sun is the most important criteria for climate change, because most of the things are controlled by the sun. vegetation, ocean, changes in land surfaces. So there are various kind of changes. Like if you see the changes in ice and the glacier like shrinkage or advance, these types of changes are. If you see the vegetation change, if there is if in any time period, if the visible environment prevails, then vegetation may change. If it's very warm for like 10 to 15 years, then vegetation may change. Vegetation may die. No, and even in the changes is ocean. If you see that the temperature or the pattern of temperature in different strata of the ocean, the upper surface and lower surface the temperature. So if the climate change, if the warming occur, then the living creature which is living in the ocean may get like if you mostly you have and sometimes we also learn the term climate variability. Always better to understand what is climate variability and what is climate change. So if you see the climate, it is a variation in state and other states and scale on the individual weather. Like mostly the climate variability is often used to denote deviation in climate statistics over a given of time, like month, season, or year, like summer season, or winter season, rainy season. These are like if one year, if you receive a very good monsoon, then another year, if you don't get, no, that is a variability. Okay, year variation. These variability occur or may be due to the natural internal processes within the climate system. Now, if you see the climate change, it refers to a statistically significant variation in either the mean state of the climate or its variability, presenting over an extended period, like even a decade or a longer, like 10 years or 20 years. That is a climate change. If the climate drastically changes in like after years, then it is called a climate. So climate change may be due to internal natural processes or external factors such as in the atmosphere or changes in land use. Even the volcanic effect also affect the climate. And if you see the definition of climate from UNFCCC defines climate is attributed directly or indirectly to the composition of the global variability observed over comparable time period. This is a recent definition. Sometimes I disagree with definition because it is written in context. It is okay. The climate is changing or affect in various time period. Even human were not there, like in PETM, that is Paleozoic thermal boundary. Also climate change. Even that time, the carbon content of atmosphere was so high, even from today's. Today we have only 400 something ppm carbon dioxide level, but that time it was more higher than today's. No, and temperature, even the precipitation long like thousands and thousands years before, but that time human was not there. Also climate change, it occurred in that time, but at present why we are worried about climate change? Because it is directly affecting us, directly affecting the human population or human society. Okay, so pictorially, weather are hours or day or month, like rain, cyclone, dry season, wet season. If you see the climate variability, it's mostly terms about the years or month wise, like El Nino southernization. But if you see the climate change, it must be occur about a decade or century level, like PTO, like global warming, sea level rises. All these are climate change effects. So climate variability looks at changes what we occur within a smaller time frames, such as month, a season, or a year. And climate change considers changes that occur over a longer period of time, typically over decades or longer. And the climate change mostly nowadays attributing to human activities, altering the atmospheric composition, and climate variability attributes to natural causes. Now, coming to the topic of Himalaya climate change. So why the Himalaya is very important in terms of climate change? Because if you see Himalaya, its extension is very wide. It has very large extension, and the recent, like 2020 definition says that the entire Himalaya, it also include the Hindu Kush Himalaya. So
Hello, Santosh sir, please unmute yourself. Hello, sir. Please unmute yourself. Yeah.
Barma. Yeah, now it's okay.
Okay,
Hello.
sir. Okay, okay.
Okay. So sometimes when I was muted.
Audible. Now it's okay.
No, this one also. I have presented this one. It was audible earlier.
Yes, yes.
Here it was muted.
From here.
Okay, that's okay. Just remind me, no? Okay, if it is muted. Okay, so Himalaya is important in terms of climate change. So if you see, the extent of Himalaya is very large, and this Himalaya also includes Hindu Kush Himalaya. So if you see H1, this includes the Northwest Himalaya and the Karakoram belt. HK2, it is mostly the central Himalaya, which also includes the North Bengal and the Eastern Himalayan region, and HK3, the Southeast Himalaya and the Tibetan Plateau. But this classification is a new classification, but I consider this is the central Himalaya, this much portion that is extend up to the eastern part of the Nepal. But Eastern Himalaya context, we usually use the west and the west Pradesh, including Bhutan. So this class, but the climate, because it is the in the northern India has this glacier, like, and this Himalaya different water basins, river basins. India, it is and other things is, even we have read in our school level that this Himalaya is important because it controls the monsoon system of the India. The Himalaya is very important to study for the climate change. And I already said this is 10 major river basin of India, like Asian river, and if you see, like Antarctic, there are two polar ice caps, no? And this Himalayan ice cap, including Tibetan Plateau, these both include a freshwater body to provide a freshwater body, and also called a third pole. This is called the third pole of the globe. And moreover, Himalayan has high biodiversity hotspot. Like Eastern Himalaya, it is a very hot biodiversity level. It has high amount of endemic plants, and moreover, it has diversity in culture and tradition and language. So all these things like affected by climate change. So this is very important to study the climate change in the Himalaya. There are reports like observed trend in mean surface temperature. If you see the Himalayan, this darker set, this one, and if you compare the Indian temperature records and Himalayan separately, the rise in temperature since 1991 in the Himalayan region is higher as compared to the other Indian region. So this is a rise in temperature. If you see the observed trend in precipitation, this precipitation has heterogeneous pattern. Like if you go towards the northwest region part of the India and the Himalayan region, these are Karakoram region, there is a trend of positive trend, like precipitation is higher. And if you see in the eastern part of the Himalaya, this portion, these are trend is decreasing. Even this color scale is also representing here the precipitation is higher or the snowfall is higher. Like if you see the monsoon wind or the wind pattern system, this region influenced by western disturbances, that is the wind coming from the Mediterranean region, and this region also influenced by southwest monsoon. But if you see the central Himalayan region and eastern Himalayan region, this region is mostly affected by the southwest monsoon season. We receive higher amount of monsoon or rain in this portion, like eastern Himalayan, central Himalayan region. And if you go towards the western or northwestern region of Indian or Himalayan region, we receive less amount of precipitation. But this Karakoram region, it has the positive trend of precipitation or snowfall is higher. That's why this region is called the Karakoram Anomaly. People are saying the glaciers are decreasing in this western Himalaya, in eastern Himalayan region. But this Karakoram region, the glaciers are expanding. The people are still doing research why this is happening. Only why this particular region has the higher, snowfall or temperature precipitation. So this is a clear term, that's a unique term as given, that is Karakoram Anomaly. So this is the trend actually how the precipitation is decreasing and increasing in the Himalayan region and different part of the Himalayan region or Hindu Kush Himalayan region. There are some projection like future projection. This is basically a concept of climate modeling. People do climate modeling based on the different existing data set, and they model and they project the present day climate scenario in future. If like certain things is like change, like if the like I have said the different component of the Earth system, no, if the cryosphere system, if you delete or input some external forcing, or if you change some parameter in climate model, and we see how it responds in future. So based on different parameter like temperature, precipitation, snow pattern, they have modeled the climate. So this up to this we are present, and this all this bar like this is called a box and whisker plot. This is showing the surface temperature is higher. It's become continuously rising, and it is compared for annual and winter season. And winter season is more alarming, like the temperature rises in higher level in winter season. Even for precipitation, it is higher, but not that much as compared to precipitation. Precipitation is mostly decreasing and temperature is becoming higher. Like snowfall, it is decreasing and it will decrease because when the temperature rises, the snow gets melted. The albedo effect is there. So snowfall is decreasing. Even in future, it's decrease. So if the temperature is rising, precipitation is decreasing, there is a sort of drought effect occur. So I consider like most important that it is drought is more like important issue or of debate. Like if drought occur, then the entire society is get affected. So it is most like okay. Now this is also future projection of different parameter like temperature, maximum temperature, minimum temperature, and precipitation, and 5-day precipitation. And these are represented in near future or future. Future means like more toward like next 100 years, and near future like 20 years or 10 years. So if you see in near future, everything is rising. So it is really a matter of concern. I hope now I'm audible, no? Please.
Yes,
Hello?
sir. Yes, sir. Yes,
Okay,
sir.
please,
You are audible.
yeah, please interrupt.
No, yes.
Okay, so this is another kind of projection. This is all based on the graded data. Like if you see for just a general information, we have station data, no? Like in like if you see the like from the Northeast India or in your North Bengal region, Siliguri may record temperature and precipitation data at any like in Siliguri main meteorological station or like Bagdogra airport station. No, they record. Similarly, in different stations, temperature and precipitation data is recorded, but we don't have everywhere. We don't have all the stations, so we have to rely on some grid data. Like these are the simulation or interpolated data. Like climate scientists, what they do, or they collect all the station data and they project in different gridded form. Like this is the Aphrodite data set. It is grid represented in like 0.5 into 0.5 latitude longitude equal space gridded. So here also the trend in like precipitation, it is the dotted region. These are trend, so it is higher. So these are kind of analysis are also important for us. Like the climate change issue also affect the snow and glacial mass. As I already said, the Himalayan is as large amount of glacier. So if the climate change, the glacial, it also affect in glacier. So this is the study of about a retreat behavior of glacier from 1960 to 2000. So here is some glacier are advancing. So here is advancing, here is advancing. Some are stable and some are retreating at very fast rate. Like here it is retreating very fast rate. And even when the glacial retreat, the glacier area also lost in different region. So here the mass loss of area, glacier area is higher in northern, in like eastern Nepal. Here in Bhutan region is slightly less, but still it is, the area is decreasing due to this melting of glacier. So all these things, all the climate change issues or climate change affect all these different parameters of the Himalayan region. So in overall conclusion, not conclusion, as an observation, if you see in climate change in the Himalaya, so it can be said like all the Himalayan region is continuously warming, no, and more than a global average. If you see the global average temperature and the Himalayan region temperature, the Himalayan region is more warmer. And if you see the Hindu Kush Himalaya is experienced a temperature rise of about 1.3 degrees centigrade during 1951 to 2014. Glaciers and permanent snow are melting rapidly in the recent year. And even the several like Hindu Kush Himalayan region glacier experienced decline in trend of snowfall and also retreat in glacier. And if you see in contrast to high elevation Karakoram Himalaya, experience higher winter snowfall, and that is shielded the region from glacial sinkage. Like I already explained about the Karakoram Anomaly. And if the like sinkage or melting of glacier or for it also affect the glacial lakes, and it increase the risk of hazard like the glacial lake outburst. You know that Kedarnath tragedy. Even we also enjoyed a movie Kedarnath, no, but that is not of this topic concern. But that was recorded like the glacial outburst. It formed a glacial lake and it is flooded, and entire that high amount of economy is lost, many lives lost, no, there. And if you see by the end of the 20th century, the annual mean surface temperature over Hindu Kush Himalaya projected to increase over 5.2 degrees centigrade under the RCP 8.5 scenario. There are various kind of scenario in the climate model, and based on this scenario, 5.2 degrees centigrade may rise. And there are some other CMIP projection is also there. Okay, so why we study climate change in Himalayan region? So these are the common things like policy in terms of policy perspective, because climate change affect directly these things like agriculture and applied sector, like watershed management, soil and water conservation, input subsidy, organic farming, horticulture. All these things are affected. Even the forest and biodiversity are affected due to climate change. Water and allied sector affected. Water quality, like water use pricing, regulation, water conservation, even a natural disaster. So if you know or if you know the future projection or like what is called the forecasting, if you have the proper forecasting, then you can control or sustainable these things, and even if affect the industry and transport. But now here again, moving to a little bit different topic, why are the available climate data sufficient to understand the climate change? I want to give a short introduction about the international chronostratigraphic chart. This is the chronostratigraphic chart that Earth is divided in like time scale, in basic time scale. Earth is formed about 4.8 billion years, and recent 1.8 million years, it is called quaternary. And at 11.7 thousand years, recent 11.7 thousand years is called Holocene. And again, this Holocene is divided into three parts: the Greenlandian, North Grecian, and Meghalayan. It comes from Meghalaya. Why it is in Indian context? Because people collected speleothem, like these are formed in cave, and based on this type specimen, luckily our institute, Birbal Institute of Paleosciences, hold this this specimen, and based on this specimen of speleothem, the recent time period is divided into 4.2 to recent present, and this is called Meghalayan phase. And you also notice, or you also read somewhere, that Anthropocene. Why Anthropocene? Where is Anthropocene, and up to in which time period Anthropocene is marked? So my question is whether we should call now the present days the period is Anthropocene, or we can mark the Anthropocene from the Industrial Revolution, or from 4.2, 4,200 years before Anthropocene. So this is always a question of debate. Again, coming to my first slide, weather and climate. Weather and climate. Okay, climate. So these are the distribution of different climate stations in the world. So if you see, this is the color scale. All the blue dots are climate station of recent time period, and red are up to back. So if you see, if you go back in time, like 1840, 1820, 1800, the climate station, the network of climate station is decreased, and recently we are increasing climate station. That is obvious. Even if you see the temporal variability or instrumental climate record of Southeast Asia, the like here, since 1951, the network of climate station are more. And if you go back in time, like 1800, 1880, it's all network become less. It's case of similar with temperature and precipitation. So where is to mark the Anthropocene in instrumental climate data? Or is the available climate record sufficient temporally or spatially? Or do we need a longer high resolution proxy-based climate model? Now it's come, if you want to study the proxy-based climate record, here is come the subject that is called paleoclimatology. And this paleoclimatology is the study of climate prior to the period of instrumental measurement. And there are various kind of sources of to study proxy data, like glaciological data, marine geological, terrestrial geological, and biological data, historical data. All these things, biological data, tree rings, pollen, microfossil, insect, corals, diatom, phytolith, and most of the glacial study. We in our institute, most of all the scientists, we work in different proxy record. So our institute is, that's why our institute named is Paleosciences, Birbal Sahni Institute of Paleosciences, because we study all these proxy records. So I'm talking about the dendrochronology, because dendrochronology is the high resolution proxy record, because it can, with the help of this, we can reconstruct climate data annually. How much time I have left? Do you have any idea? Can I take another 15 minutes?
Hello, Tapas sir. Hello.
Hello. Can I take another 15 minutes?
Yeah.
Hello, Papa. How long will he continue?
Okay.
Maybe 5-10 minutes, no?
10
Yes,
minutes
sir.
is okay. 10 minutes is okay.
Yes, sir.
Okay. So if you see the tree rings, these are called the tree rings. It forms in annual, like early wood and late wood combined as one ring. So these are the annual rings, and these annual rings can be dated. Like the rings which is near to bar as the recent year or year of collection, and the rings which is toward the pith, it is the older, like this may be the 2018 rings, 18 years, and this is the. So based on these tree rings, we reconstruct climate. And there are processes. These are the tree rings, how we collect, and these are the rings, tree core sample. Then we count each rings. Then we measured each ring. These are the graphical presentation of each rings, and we measure each ring, and then we plot it in one graph, the average of this all tree rings pattern, and then we correlate with the climate. And whatever significant season we get, that we reconstruct. That here, like red is the instrumental data, and this blue one is reconstructed data. These are the method, like the same method which I already explained. These are about the calibration and verification of trading with record with observed temperature or any climate record, and these are the reconstructed. Like see here. Like here, climate data, the red one, here in the background we have tree rings, okay, and the red one is the climate data. So if you have climate data up to this much, like 1900, then this blue one is the reconstructed data that is based on the tree rings. Why we do reconstruction? Because to get a proper climate dynamics of the region, we need longer climate data. Whatever study I have shown, that all are based on the instrumental record. But if you have longer climate record, then it will represent much more in clear terms the climate dynamics of the region. So I'm presenting shortly a few climate study, like this is from the Western Himalaya, precipitation. This we have carried out in Kashmir Himalaya, and even if you see the Kashmir Himalaya, again the network of climate station, it is all decreasing. We have only one station, that is Srinagar station, back up to 1819. So these are tree ring sample we have collected from here, Pahalgam. Me and my one of my student, a PhD student, Uttam, he is also listening this talk, and we collected and we have carried out this study. And these are the chronology. These are based on more than 100 tree ring samples. Like these are from two sites, okay, and see how this perfectly matching with each other. Two sites but matching perfectly. And this we have compared with the climate data, and climate record we have seen the April, May, June months that precipitation, it is a positive signal, positively correlated. So based on that positive correlation, we have reconstructed this precipitation signal up to 1720. Okay, 1723, I guess. And we have compared, we have like checked these all this low. This is triangle which is downward. All these are the drought years, which is perfectly matched with the all drought records, and these are the flood records. And even we have compared with other existing instrument like reconstructed data, and it is also matching perfectly. And we do some spatial correlation. Spatial correlation is we have the reconstruction here. Then how this is, how far, how the extension, how far it is extend, it is possibly capturing the same signal that we do with this spatial reconstruction. This is the temperature. This is also published in Climate Dynamics, and this is from also from same region, but these are based on the blue pine, that is Pinus voliciana tree ring. This is climate, and these are the tree ring chronology, and these again compared with the temperature and precipitation record. Here the Pinus voliciana tree is positively correlated with winter temperature. So if we have reconstructed in that region, we have only the temperature and precipitation data available only from 1979 to 2013. But with the help of this tree ring, we have extended that temperature, winter temperature record up to 1850. And if you can see, in terms of this entire 150 years, the recent temperature is rising. So we can say with the help of this tree ring based reconstruction that the climate or the temperature of that region is higher or continuously rising as compared to 160 years. So for this we use tree ring, and we can get the longer climate record. And again, it is compared with other region reconstruction. Everywhere it is all rising, like and region. Again, skipping, and this we can see how this climate is controlling by different patterns. Here the oceanic influence is there. Oceanic influence is represented here. We also study drought. These are the study from Nepal, Himalaya. Again, the same reading. It is also based on Pinus wallaceiana, and these are collected from the western part of the Nepal, from Dolpo, Himalaya, and these are the climate data of present. This is only available from 1957 to recent. Then these are the chronology of trading chronology, and again compared with the different like temperature, precipitation, like and spatial correlation. These are the gridded form of data. So here is the trading chronology. It is compared with this gridded climate data, this data, all. So we observe it is responding negatively with precipitation at a temperature and positively with precipitation. That means there is some kind of moisture level is playing important role for the growth of this tree. That's why we are again correlated with drought. So we got the positive result. That is drought or the soil moisture is prevailing or controlling the growth of this tree. That means we can reconstruct drought record up to 1700 AD. So these are the observed and estimated drought record for instrumental period, and these are the reconstruction data. If you see here, these are the mean line, this is mean. So this is lower, and here again it is declining. So we can say the drought is declining, negative is occurring in nowadays. So this we can say the more drought period are prevailing or prevailed in recent decades. Again, we checked with other regions, and it is almost showing similar effect. These are some case studies from Eastern Himalaya. Even the people believe that not believe, it is actually the conifer taxa only produce annual rings. But we have carried out this trading study of tuna ciliata from my hometown Kalimpong, and this is the hometown work from home work. Why? Because this is from my hometown work, and this is me, and this Nivedita Mehrotra, she is my wife. So we carried out this work jointly in our hometown region, and we saw the even tuna ciliata, it is almost 160 years old. Okay, and this is broadleaf taxa, and this is correlated with temperature and precipitation data. Here also in Kalimpong region, the winter temperature is raised, and it is perfectly matched with the growth of the tree. Okay, so all the temperature, winter temperature is rising in this recent period. Even the volcano effect tree pattern and temperature. When the volcano erupts, it gives the huge amount of sulfur dioxide in atmosphere, and a thick blanket of sulfur present in the atmosphere, which protect the penetration of sunlight. Then the temperature become lower. So same thing is happened in history also. This is the largest eruption of 1850 eruption of Mount Tambora, which is the largest volcanic event in the last millennia. This is from Indonesia. It occurred in 1815 AD, and after that the temperature is become lowered for almost 10 to 15 years. And even in history, in other year also, when the volcanic erupt, the temperature become cooler or lowered. So this is the Great Britain bristlecone pine. This is still growing. It is still living, and it is 5,068 years old. So you can imagine, if you reconstruct the climate history based on the trading, we can get a climate history of entire 5,000 years. Even in Himalaya, like cedar, hemlock, juniper, chelgoza pine, these all are growing more than 1,000 years. So we are getting climate history of 1,000 years in the Himalayan region. Trading, we have wide application. We can reconstruct temperature, precipitation, drought. Even it has application in forestry management. We can study past earthquake, history of stream flow, landslide event, volcanic eruption, glacial history. There are wide application. Recently, we have carried out work in Bhutan, 2018. We have seen that these trees which are growing near to streams are got affected, and we have sampled. And if you see in enlarged view, this is one annual like true rings, and this is the false mark, which is a flawed scar mark, and this is for year 2009. And 2009 there is a great cyclone was came and affected entire eastern Himalayan region, and this is a storm track and integrated water vapor transport area, and we have collected many trees, and we got all the rings has 2009 scar. This is flood scar. So you can build a flood history based on training, and that also contribute in climate science or climate change study. So the points to ponder is that are the Himalayan or Indian long-term climate variability and or change following the global trend? Are the global trend and the Indian or Himalayan is following the same global trend or not? That we have to think about it, or we have to study. And the climate variability has its own local or regional scale trend. It doesn't seem that Eastern Himalayan climate is same as compared to Western Himalaya. And there is only, or we have to check whether only the extreme event is following the global or hemispheric picture or not. That we have to check. And do the forcing factor vary locally, regionally, or hemispheric globally? Because like the Tambora eruption, it affects entire globe, but in some area it has not. There is no effect. So do we see the entire frame or just a picture, or we are finding the needle in a haystack? So the few take home the spatial linkages in terms of climate calibration and global teleconnection pattern are needed in the present climate change study, and the past climate reconstruction are needed in both temporally and spatially scale. Like temporally in like 1000 years or 1500 years, that we have to see. We have to see in a spatial scale. It is not possible to study in one particular site and we'll project that no, this is the climate of the entire region. No, we have to check with the spatial scale. We have to study in broader scales. And like in Eastern Himalaya, we have to study entire Western Himalayan region based on the spatial scale. For a broad, and this is required for broader perspective and to understand the climate dynamics. And now the time is bring together the policymaker, that is a climate policymaker or the governmental policymaker and climate researcher together. So thank you very much, and this is our institute for future. Must opportunity to talk in front of you through a webinar. Thank you very much.
Dr. Papa?
Yes, sir.
We are already in shortage of time. Only one or
There's lots of question, but you select the one or two.
yeah, you select. Yes. Papa.
Okay. Now, is the climate disease? No, I don't think. Even it is not advisable to tell that climate is changing due to the corona. No, I think it's a variability. Climate varies, no? What
Hello?
NASA got to, yes?
Yeah, Papa, do you have any question? Hello?
Hello, sir.
Yes, sir.
There is a question. Do you think that climate change in the Himalayan region has anything to do with the recent frequent occurrence of thunderstorm and cyclone in the lower Gangetic plain?
Climate change in Himalayan region does affect the cyclone and storm.
Thunderstorm in lower Gangetic plain.
No. These are all actually the natural climate system, natural variability. So I don't think it is related to the climate change. Because but it affect due to the like temperature, like the oceanic effect. Like if there is a special pattern change in the ocean, then it affects the local climate. So there is a teleconnection actually. So, and these are not a long-term effect. These are the temporary, like even, I can say that, no, it's not actually related to climate change. Storm and cyclone, it is actually due to the turbulence or changes in oceanic pattern.
Okay, thank you, sir. The organizer of the colloquium would like to thank Dr. Shah for his enlightening presentation on climate change in Himalayas.
Thank you very much.
As he pointed out, climate change in the Himalaya, indeed his impact on large part of the Indian population and climate. In his lecture, he discussed about the different strategies by which climate change can be studied. And we learned lots of things from your presentation. Thank you, sir. Thank you.
Thank you very much. Thank you very much. Namaskar.
Okay, thank you, Dr. Santosh Saha, for your time and deliberation. Now we are ready. Yeah, we are already running shortage of time, and then we'll just directly pass on to the next speaker. And our next speaker is Dr. Silas Ranjitka. We are sorry, Dr. Silas, that we kept you waiting. Manjil.
It's okay.
Hello.
Yeah, thank you.
Yeah, Manjil Gupta, would you please introduce Dr. Silas?
Yeah, thank you, Dr. Thapa. It is my honor to introduce Dr. Silas Ranjitka. He is currently ecologist and lead researcher, Tree Diversity and Domestication Unit, World Agroforestry, East and Central Asia Regional Office, China. He did his BSc from Trichanda Multiple College and MSc in Botany from Tribhuvan University, Nepal. He was awarded PhD from the Chinese Academy of Sciences, Kunming Institute of Botany in China. Dr. Ranjitka is a creative and innovative ecologist with more than 15 years of experience in the field of mountain ecosystems, biodiversity, and ethnoecology. His expertise is in the planning of the agroforestry system using spatial and land suitability analysis. He has constantly worked with farmers and policy-level stakeholders to promote agroforestry, economic commodities, developing agribusiness. He has worked with diverse groups of people on various commodities in China, Nepal, Bhutan, Pakistan, and India. He has worked on a number of research projects as lead researcher. Dr. Ranjitka has published over 35 peer-reviewed articles in international journals, 9 articles in national journals, and 1 book on commercially important medicinal plants of Nepal. With this brief introduction, I invite Dr. Ranjitka to deliver his talk. Over to you.
Thank you, Dr. Sarkar, and thank you, Kishore. Yeah, I'm trying to share my presentation first, and then I will discuss about it. Can you see my slides?
Yes, sir. Yes, sir.
Visible, sir.
Okay, thank you. Thank you for introducing me, and I really appreciate presentation from the Santosh that saved me a lot of time to talk about the climate and climate change, because my topic is also related with the climate change. So while he was presenting, I was just editing my slides and reviewing certain parts that I think are not necessary. I should repeat. So my presentation, it is mainly focusing on the hybridizing scientific and indigenous knowledge for climate change adaptation. So I will be talking a little bit about the climate change adaptation mostly about the ethno-ecological part. It includes many components from social aspect to the ecological modeling aspect. So huge part of research is compiled within this presentation. That's why if there is kind of like a information gap or the information not sufficiently addressed in this diverse group of let's say webinar attendant, I really would like to request to send question to my personal email that I will give you after my presentation. So without delay, I would like to present. It is about the ecological calendar in the Himalayas and the Panis, but I would like to mention it is in the Asian highlands. So what is ecological calendar? First, let us little bit talk about it. It is not the new concept, but introduced as a new term recently. It is a knowledge system. We can say it is a knowledge system used to anticipate and align livelihood activities along with the seasonal changes. So we know like we have different season in our region, starting from spring, summer, we'll have a rainy season, we will face autumn, winter. So along with all this season, what kind of activities are carried out and what kind of adaptation or the adapting measures people are taking since they are residing in a certain places. That is the key for the ecological calendar. Can you hear me? Hello?
Yes, sir, you're audible. Yes,
Okay.
sir.
Yeah, because I feel very blank, that's why. Anyway, so by tracking this seasonal development, we call them ecological timekeeping or the ecological timekeepers. This system provides communities the ability to anticipate agricultural activity as well as cultural practices. So this knowledge system of ecological calendar, it includes astronomy. Before Santosh also present, like sun is very important component in the climate. So astronomy things are there, meteorology and phenology. And all these knowledge are connected to one system. That is how these calendar are developed. So when we talk about the ecological calendar, we talk about different cultural band that include the various festival, religion, agricultural band that include how trees are grown in the local area, what are the fodder and the food resources, phenological band that include the life cycle of animal, plant, even the insect are included. Then the art band that include information coming out from the glacier, snow, permafrost, river, all these geological changes. Then astrological band that include sun, moon, or the star band, which is part of the astrology itself. So example of ecological calendar in the region. If we if we try to dig when these kind of calendar come in existence, we'll find something like a 5,000 years ago in the eastern part of China. There is a system of keeping this kind of schedule, which is documented formally around 2,700 years ago in the Yellow River Basin, which is very popularly they call as 24 solar term, as UNESCO already listed it in the World Cultural Heritage, I should say. So this 24 solar term, the Chinese calendar, early Chinese calendar is one of example. Then another one is calendar of human body from the Pamir mountain, which is also very, very old, and it exists from very long time. So what is in the 24 solar term? You can see 4 squares here. These 4 squares represent 4 seasons, starting from spring, summer, autumn, and winter. And what kind of activities, what kind of observations are made by the local people, indigenous people, are documented here. So each solar term divided into 3 periods, they called. So there are altogether 72 pentads in the year. Each pentad consists of 5 or 6 days, which are named after phenological event. So all these terms, these are indicating certain phenological indicator or the phenological event. It doesn't matter whether it is related with the plant, whether it is related with the bird, whether it is related with the animal or even insect. These are included here. Then a solar movement is included. Meteorological knowledge is included here. Then the agriculture activity are here. So if you check at the left corner here, talking about the spring, they started mentioning with melting of the like ice layer in the frozen lake and reappearance, I should say, reappearance of a fish, or the fish, they are surfaced, because before that the lake is frozen and mostly these fish life, they are at the bottom, and when spring arrive, fish are now visible. There might be the flying geese, they can notice them. There might be the blossom of certain plants, especially cherry, which is very popular and very important in that part, like Japan and eastern part of China. So these are included here, and it ends with telling about the how changes occur at winter, during the winter. So it mentioned in the last part here, it says like these geese, wild geese, they start to fly to the north, fly to the north. Then magpie start to build their nest, because now last part of this is kind of like a link with the early part of the spring. Similarly, like hens start to feed, this means wild hen, they start to feed their chick. River and pond. Sorry? So,
No, you are audible.
Okay. Sorry. Yeah. So anyway, the lake, it is again a river and pond are frozen. So these events are mentioned in this calendar, which is a kind of documented in the very early time. So another example is a human body calendar from the Pamir. So these calendar are mostly driven based on the observed cue in the environment that spread across calendar of the human body. So these are local timekeepers name each new season development after a part of their body. So mostly it is begin with the toenail, then move upward, reaching up to the head. And the arrival of the head, actually it coincide with the end of one season, let's say spring, and the pause continues there. Then when they observe first cue of another season, let's say summer, then they start to recount, and this time it is from head toward down. These timekeepers rely on the most of the events such as the flower bloom, arrival of the migratory bird, movement of the fish, breakup of this lake ice, which is very similar as in the Chinese calendar. So these indicators are kind of like telling, okay, now seasons are changing, and we should keep these in record to adjust our agriculture activity. So this is, all these calendars are mostly and mainly
Hello, sir. Please unmute yourself. Somebody.
Sir, kindly unmute yourself.
Can you hear? Can you hear me now?
Yes, sir, you're audible.
You're audible, sir. Okay, now we also got the video. Okay.
Yeah. Sorry. So I was talking about this slide about the human body calendar. So mainly here we talk about like a flower bloom, or the phenology of flower, arrival of the migratory bird, movement of fish, or the breaking of a lake ice. These are the seasonal indicator, and based on that, these calendar are kept, and this information is mostly kept for purpose of the smooth agriculture. So constructing of ecological calendar. That is our main concern now, because this is a kind of a traditional knowledge, and we really want to see if it is still in existence, or and if this information is really useful for local adaptation in context of changing climate or in context of changing environment. So the main thing is, these calendars are no longer widely used, because now we have a very good timekeeping system. We have a modernized meteorological station to inform about the climate, weather of a local area, and also there is a lot of agriculture services. But still, these calendar could be useful. That's why we focus on this ecological study. So what is our consideration was memory of their uses. These calendars uses still survive, and the word from the calendar marking specific ecological event in local language are still in use, which could be very useful for the local climate change adaptation practice. Hence, construction of the ecological calendar is possible, and that require proper identification of seasonal indicator and various other factors that we will discuss. So these are the key component. You can see publication from the Kasham et al. 2018. He's an American professor and leader of this ecological calendar project in our consortium. Anyway, this construction of ecological calendar this require seasonal round of agriculture and social activity. How local people are currently practicing their agriculture, or mountain people are currently practicing their agriculture practice, that we can call a seasonal round. Seasonal indicator and their connection, which could be fit into, again, seasonal round. Then consists of the use of specific seasonal indicator. What are these seasonal indicator, and how they fit, and how they are frequently used by local people or the different communities in the mountain, that are also important. Then the reliability of phenological indicator, these are also important to regulate and to relate it with the climate, because phenology are very clearly related with the climate change, and it can be say it is a first indicator of a changing climate. Then why climate matters in this all issue? Because climate change could disrupt the seasonal marker. Second, a seasonal marker help the mountainous community to maintain their routine, their schedule. Therefore, disruption has made them uneasy. A shift or a shifting in the climate is disrupting not only their subsistence farming, but also the herding and the unique way of tracking the time that they have developed since a long time. So if we check out these highlands, Asian highlands, we can see temperature rising faster in the higher elevation. So glacial is melting. We also hear this in the earlier presentation. And the rate of the glacier melting is higher than the global average. So lost after permafrost, mostly in the Tibetan Plateau. Impact on the ecological interaction, such as the shifting of species range, invasive species and pests are incoming. So these are a kind of ecological impacts currently happening, or that might happen in context of a changing climate. Disrupted agriculture cycle, that includes
So you have been muted once again.
Please unmute yourself, Alice.
why it is happening.
It's okay. It's okay now.
It's okay. It's okay.
Okay. So now just I am talking about impact of the climate change. You can see this publication from my boss, Jian Chishu, which talk about how vegetation shift because of changing climate. When we projected around 5 degree centigrade warmer than the current climate, how vegetation will shift in central Himalaya. It is a cross-sectional view of it. It will have a big impact on the community and the livelihood. So from this point, we could ask how climate science work with indigenous ecological knowledge or the indigenous community to document this ecological knowledge, to update and recalibrate the calendar to anticipate climate change. So this is a big question we put to work on this concept. But there are several other questions, like how people in different places adapt to local climate change, how traditional knowledge help people to adapt local climatic variation, how do indigenous people operate their seasonal activity, do they have any timetable still in existence to conduct their seasonal activity, and how people are using the resources around them, and what kind of ecological knowledge they have. So these are the questions we put together, and we hypothesize that we can generate a hybridized knowledge system that include a scientific information from different branch of, let's say, meteorological science, or even like ecological modeling or the phenological modeling, and how that can be integrated with the ecological knowledge from an indigenous community that live in a certain area, certain mountain area for a very long time, and what kind of seasonal practices they have. So using this all set of information, we are trying to develop or reconstruct an ecological calendar that could be used as a climate smart knowledge for the local adaptation planning. So this is the main theme of the work. So I'll directly now move into the different results that we obtain, different findings, and a little bit we'll also discuss on that. So ecological calendar cues, from where we can get the information, where we can get the fact for that. From ethnoecology and traditional knowledge, the first important aspect of getting the knowledge is traditional knowledge from indigenous people, which are very important to map and monitor ecological change and improve scientific study, because we are trying to get a certain knowledge, certain information based on the certain local area, and in that area these indigenous people are living since very long time. They have very good knowledge about the surroundings, they have a very good knowledge about utilizing the surrounding resources, observing the resources. So they are a kind of a source of very good information. That's why they are important, what they can provide. So these indigenous people, we found that still they can recognize their own indicator of environmental changes. They have certain kind of like indication, or they have certain kind of understanding of certain change in their surrounding is indicator of certain kind of changes is happening, or soon it will happen. So that is kind of like a change in the water volume in the major river, in the high altitude especially, snowfall and snow melt, arrival or the flying away of the migratory bird, arriving of the insect, greening of the rangeland. If you see this picture, you can have a different tire, like a snow-capped mountain, snow change can be clearly visible. These pasture land where greening or the graying could be clearly visible. Similarly, like in other adjoining area, beginning after flowering, that could be clearly visible. Even like a migratory bird arriving or the insect arrival, that could be very easily notable. So the people, local people or traditional people, indigenous people, they have these kind of knowledge preserved in their pocket. So that is very important. So seasonal observation, we found several indicator species during our study from different part, like wagtails consistently throughout the Himalayan region and the Barma region. Wagtail are very important indicator of certain change in the season, or the certain starting or carrying out certain activities, such as the sowing in the Barma region, like yogurt making, butter making, and this worm, it is one kind of food in Barma region. So during the arrival of this wagtail, two species of wagtail, inform them, okay, this is time of making or doing this kind of activity. Similarly, like this upapa, they are indicator of arrival of a spring, especially in the Pamir, whereas the same species in the southern part of Himalaya is indicator of arrival of autumn, because their migration time fluctuate with this season. Similarly, the flowering of the Cecum species, dandelion and the daisy are also important indicator for spring and summer. These are also quite consistent across the Himalayan and Pamir region. So this is one example of how pastoral activity are carried out in the Pamir region, depending on the ecological indicator. So in Pamir region, they move from one pasture to another pasture. They are mostly kind of like a seasonal migratory. They need to take their animal from pasture to pasture, depending on availability of the fodder for its resource. So spring pasture, you can see temperature. This is one important indicator. They perceive warm or it is cold, right? Similarly, like a soil condition, if soil turns soft, so turning of soil into a softness means it is time to move to the spring pasture. And another important indicator is grass around 2 to 4 centimeter, as indicated by this old guy here. He is showing this much. Similarly, color of the grassland, it can be visible from a distance, and the snow melt, this is important, and the visibility of ants, insects such as ants, these are also important indicator of moving to the spring pasture. So from spring pasture to summer pasture, again, temperature, height of the grass, snow cover, decrease in the summer pasture, water resources in the valley, the river get bigger in the valley is indicator to move to the summer pasture. So similarly, there are several indicators to move into autumn pasture and the winter pasture. So this is one example from Pamir region. So based on that, we can develop a kind of a seasonal round. You can see this seasonal round, we can put a different season, timing of different activity like a photo collection, timing of animal to behave with their changing in the period, like a hibernation during the winter for bear and marmot, like wagtail, this white wagtail and citrine wagtail appearance during the spring, availability of a wild plant for wild plant here mostly means wild edible plants for collection for human purpose or for even for the cattle. Similarly, cropping season and the fruiting season. These can be included in a one small round, this seasonal round. So we published this result in a publication in 2018 in sustainability. You can access this if you want to read. So second Q, it comes from the phenology. So phenology is very important, as I mentioned earlier. It is an indicator of first indicator of a changing climate. In phenology, what we can see for the climate signal is like a emergence of a leaf, leaf expansion, bud burst, flower emergence, full bloom, fruiting, leaf senescence, leaf fall. These are the events which tell us which can be correlated with the temperature or can be correlated with the local weather condition to check how weather condition or the local weather condition is affecting these events. Similarly, it can also tell us about the ecosystem indicators such as the start of the growing season, end of the growing season, even the growing period and the dormant period. These are very important indicators that tell us how in different these season, these events matches or how these are changing with changing climatic condition. So when we talk about the phenology with the seasonal cue, in a normal year, we can expect like certain flowers are blooming in the May, seeding time and flower maturation time in September. So if there is a early rainfall, then we can expect little bit shift in the phenology. But using the celestial calendar, we can just find like, okay, we can observe this is flowering is earlier, but our sowing time, because in the normal year we do it in May, we will be again stuck with the May for that. And for like harvesting or the maturation, that will have a certain problem. But if we follow seasonal cue, then the flowering or the blooming of certain wildflower is indicator of, okay, this is time for sowing. So it is kind of linked with observation of ecological indicators that will help us to manage our agriculture. Not necessarily, this is the example from the sunflower, it could be certain crop plant that is important. So phenology from historical specimen. We did a lot of phenology work to gather the information, how phenology is shifting in the region, how phenology is related to climate. In that series, we published one paper in the PNAS using around 10,000 plant species, 10,000 herbarium specimen deposited at different herbaria across the world, concentrating on 27 species of rhododendron. So flowering time of rhododendron, these Himalayan rhododendron respond to annual temperature, fall temperature, and elevation. These three things we find there. And mostly specimen recorded from this 1950s to mid 2000. So 1.4 days later per 100 meter. That means flowering delay by around 1.4 days when we move around 100 meter upward. So similar thing we try to check at the field using a elevation gradient study from around 3,000 to almost like a 4,000 meter. This is carried out a certain time during 2010 and 2013, and these are the publication where I lead the paper. We found around 4.3 to 5.8 days delays per 100 meter for the beginning of the flowering. So from earlier period, you can see around the 2 days per 100 meter. In the latter time, it is almost like a 5 days delay per 100 meter. So it is in the higher elevation, it is probably a little bit kind of like a warming is delayed compared to the lower elevation where it is quite faster. So another example we have from the crop tree. We did similar study in the apricot phenology from the Gilgit, Balochistan, Pakistan. This is, this should be apricot, not apple. So flowering in the apricot delay around by 3 days with every 200 meter elevation different. We have a monitoring plot at a 200 meter elevation difference, and we found around 3 days delay with rise at the elevation. So this is the result of the partial least square regression that shows how environment affect on the phenology of apricot. Here we have a variable such as the temperature. Mostly we use from August till April because this is the season of kind of like active period for this apricot. After April, it will enter in the phase of the fruiting, so we ignore that part. We are only concerned about the flowering part. We found that winter and the spring temperature are mostly significant, these green bars, and the red bar, they are non-significant one. Mostly during the winter and spring, temperature warming could advance the flowering condition. That means flowering will get earlier if temperature rise during the winter and the spring. Similarly, elevation also have a positive effect. Elevation have a positive effect, that means a delaying effect, and different soil factor also have a delaying effect on the phenology. But the important thing here is temperature, warming temperature have advancing effect. So that is the key point from the phenology here. Then another cue, it comes from the crop season, yield, and the climate change. So if we plot growing degree day. We develop a growing degree day indicator and try to link it with the wheat production, wheat yield. So we developed it for several decades and divided it into 6 these categories, and found that in the recent time it is already like growing degree day accumulation or heat accumulation, we should say. Heat accumulation is the amount of heat that is required for any crop to get maturity or beginning its growth season. So beginning of season as well as end of season, it is getting earlier. Beginning of season, it is around 11 days, and you can see end of a season, it is almost by 30 days advancing. So that means growing season is getting shorter in the current period, which could be good for some crop and may not be good for some other crops. So if we check this growing degree day pattern with the local practice, this calendar thing, wheat and barley seasonal like cropping thing, it is from the local information, it is from the local practice, and this is from the climate model. It shows like wheat, they already start these days from late March and they keep until they harvest until middle October. Barley is quite shorter than wheat. Around it take 114 to 213 days in the field, and this growing degree day chart also shows us right now it is getting shorter and earlier. Planting could be done earlier. So earlier, when somewhere here, that means around at the mid of March. So they already start from the late March. It is quite coincide with the this chart. So these are just the result of these two comparison. So we did again this partial least square regression to check impact of these two climatic indicators such as evapotranspiration and growing degree day on wheat crop, and we found that evapotranspiration mostly have a negative effect that could reduce yield, which means if warming continues and evapotranspiration is higher, then yield of wheat will reduce. That is one important finding. And another one, growing degree day. Currently, what is because of like warming, how much heat accumulation these crops are getting in this mountainous region are really good for the yield, and potentially to some extent with the warming condition continues, yield might be better for the wheat. Similarly, here we plotted it. The wheat yield that is we collected from last 25 years against the evapotranspiration and the heat accumulation. It shows around at this point, you can see around at the 2100 centigrade heat accumulation and lower evapotranspiration, yield could be very high. But if evapotranspiration exceed around 380 millimeter per year, and if heat accumulation exceed around 20, let's say around 2200 degrees Celsius, then that is kind of like a extreme point for yield. So beyond that, or if evapotranspiration increase above that, yield will decline constantly. So this is a finding from our this crop model yield, which is now under the review. Now another information comes from climate. We already heard about the climate and the climate change in the earlier presentation, so I just skip a lot of part here. Just want to say like there are a lot of climate change scenarios at the present, and we assemble those data. Around 19 models are assembled here, and based on that, we found like there is 1.6 to 2.5 degrees Celsius increase in the mean annual temperature by 2050. Similarly, talking about the precipitation, there might be reduce at some places and increase up to 375 millimeter by 2050. This is again based on the ensemble of, or you can say averaging of this IPCC AR5 scenario 19 model available for the modeling purpose. So what does these temperature and the precipitation change indicate? Definitely a change in the vegetation might be there. So there is a paper from Nature et al. Published in 2013. We talk about global environmental stratification. This is kind of a new system of dividing ecosystem, we can say, based on the climatic information. So what climatic information used here are the seasonality, temperature seasonality, annual temperature, aridity index, and the evapotranspiration index, potential evapotranspiration index. So these 4 indicators are used here, and based on kind of statistical work with these 4 indicators, they have divided world, let's say, vegetation into or the vegetation zone into different these strata, or the different stratification is carried out. So you can see from A to R, these are the current system they proposed. These are the older ecological zones. So we use this and developed it for Asian highland, and found that there will be a lot of change in these bioclimatic zone. We can call this bioclimatic zone. There will be upward shift of these bioclimatic zone around up to 423 meter in average. So a lot of zone will kind of like, you can see, change a lot, up to 96%, and lowest one, I think it is 9. This will mostly gone, and the new, mostly the extreme hot and arid condition, hot and dreary condition, this will arrive in the Asian highland, and where changes will happen, you can clearly see. And this in the mountain region, if you see in the mountain region where hot and mesic region are there, this will mostly be changed into extremely hot and arid or the xeric condition. So that would be really devastating for the lot of agriculture, but also could provide opportunity for some other crops, especially in the high altitude, in the Palmer region, or in the, let's say, I didn't mention about Karakoram here in the Hindu Kush and the Karakoram region, and also in the high elevation of the Himalaya, including the Chinese mountain region. So climate change induced shift of the key species and agriculture. So how change in this bioclimatic zone will have an impact on different species and agriculture, that we will talk a little bit here. I used the rhododendron arboretum as a indicator or the keystone species to check how its niche will shift because of change in the climate and how it is currently distributed, because this species is distributed throughout the Chinese mountain region, Himalayan region, Karakoram, even it reaches border of the Pamir. So I said this species to Moru, and we found in context of climate change, you can see mostly it will invade alpine ecosystem, and it will lose habitat in the temperate zone, these blue areas. Even you can see in the Meghalaya, in the Arunachal, even in Bhutan, here in the Darjeeling, throughout the Nepal, even in this lower part of Yunnan. Interestingly, this species is also present in Thailand, Vietnam, and Laos, and most of its ecological suitability will be lost by 2050 in these regions, and it will mostly shift toward Tibetan Plateau by 2050, invading the alpine ecosystem. So this is an example of how change in the bioclimatic zone will lead to the shift in the keystone species in the region. So if we talk about the crop, Here is another paper we published in PLOS One, again using the similar model, and we found that you can see peak elevation where most of the current plantation is there, and most of the suitable niche is there, will shift almost by around 300 meter upward. Here you can see in different scenario. It is consistent across the whole scenario. Even their rate of climate change is consistent around that distribution across all the scenario. You can read this paper. It comes from the another model called the climate analog that we carry out. So climate analog is a kind of a model which is mostly done to search the future climate in the present. So just the tag would be the search for the future climate in the present. Here, just briefly, I would like to mention present climate of any study area can be, future projected climate of any study area can be found in any of analog site at the present. Based on different latitude or based on different elevation, this could be possible. And searching that is a kind of a modeling work, statistical work carried out in this analysis. Climate in the present, we targeted one area called the Yunnan. It is in the Yunnan province of China. Here you can see how current climate condition, minimum temperature, maximum temperature, and precipitation in the green is in this area, and the red one is a projected climate change. We search where similar climate could be potentially located at the current in the present scenario. So we find almost similar climate. You can see minimum temperature are almost similar, maximum temperature a little bit deviate, precipitation almost similar. So such location we find to check what kind of adaptation practices are carried out in these target or analog sites. At climate analog location, a manifestation of the projected future for a given location can be visited and investigated to compare existing farming practices and the ecological calendar. So this is important information because we don't know what will happen in the future. We can just have some projected scenarios, but we can see that projected scenario, similar thing in certain other places. So the information from that other places could be very useful for adaptation in our current site. That's why this analysis carry very important position in the research. So for that site, we located like a best analog site, the green one here, a small one, which is just 250 kilometers away from this study site. So it is quite close. Just the elevation differences might be there. However, you can see the red, all these red or the warm color, they are also the climate analog site for this particular study site. That means we have a lot of options now. The practices carried out in this part, or even in this part, or this in the Bhutan, or even Nepal, that could be also useful for adaptation practice in this particular site in Yunnan. So the climatic distance, lower the climatic distance, similar practice for the future adaptation could be found there. So what information we can get from these analog sites? We can get crop yield or the crop yield potential information. We can get information about the seasonal calendars or the seasonal pattern of these cropping or the crop calendar, we can say. So these information are important. So we check one of our analog site of this one. So analog site is somewhere here, this one in the central Nepal. So it is in the central Nepal. We check yield potential for several crop. I present here 3 for maize, potato, and wheat. The results are matching with the finding from the household survey at this area in Nepal. Farmers are already replacing wheat because of low production. If you see wheat, it shows this red dot is a current potential yield, and these box plot show the potential yield in the future. Most of the future yield is below this red line, which means in the future, wheat yield could be reduced much in this area. That means wheat would not be a suitable crop to adopt in the our study site, because in the warming condition in the future, this will have a lot of problem with the yield. So replace and reschedule the cropping time as a crop management part would be the useful from this information. So similarly, another example, it is from the Dros, Pakistan. It shows potential yield of maize would increase by 2050. This is not the analog site for the site in Yunnan, but this is a independent, different part. So just I would like to show this chart, because earlier we see much reduction in the wheat, but in this case we are seeing it is really good. So current yield is somewhere here, this green dot, and most of the green dot in other scenarios, climate change scenarios, are much better. That means in the future, there is really good opportunity for high yield of this particular crop, maize, in the Dros area of Pakistan. That's why adopting this crop and the crop calendar from the analog site would be really beneficial for the farmers of this Dros. Hence, locating future climate today can provide opportunity to enhance knowledge of adaptation capacity and supporting the identification of appropriate interventions. So it provides good information for reconstruction of the ecological calendar. Now, combining information from all these information, source of information, such as the ethnoecology, phenology, and the different modeling work, what we can get is certain kind of integration of all these kind of information. So this is an example from the ecological calendar from southern aspect of Himalaya in the central Nepal. So this is done with one of ethnic group called the Thakali, how they observe environment, what are the indicator species they identify, how their agriculture activities are linked with these seasonal indicator, how they harvest any of a wild species such as like Ophiocordyceps sinensis or the Merkela esculenta mushrooms that are available during certain period of time, period of year, similarly medicinal plant or the edible plant, or even how they observe migratory birds, how they observe wildlife, all these are included here. And crop calendar is at the center, and the link between them can be key to the ecological calendar. So how combining this? So in the software, in the computer software, we can combine this information carrying certain within the certain time period, how changes are occur. Let's say within the 2 weeks timeline, what kind of events are happening and what kind of knowledge we got from local people that could be fit. Agriculture cycle could be fit there. Meteorological cycle or meteorological data can be incorporated. So indicator species included.
Excuse
and
me.
Even like a, yeah, please.
How long would you take now? How long?
Sorry?
How long would you take now? From now?
I have,
You need.
let me see, 5 or 4, yeah, just 5 more slides.
Okay. Okay. Thank you. Thank you.
Yeah. Okay. Let me continue. Okay. These are how we are constructing this calendar, and all this can be incorporated for a yearly calendar that include different season. Here is example from the spring, 3rd and 4th week of March. What activities are here? What kind of indicators are present? What kind of agriculture activity could be important? How they are linked with the local weather system? and what kind of indicator from the phenology are there. So all these can be linked here. This provide a base for the ecological calendar, and why this is important and how we can adopt it. So let's say we are working at the bioclimatic strata 1 at a lower elevation. What we documented here could be really useful, and the knowledge transfer can be carried out in the higher elevation settlement. So the knowledge system that is developed in the lower elevation could be useful for adaptation planning in the higher elevation. So this elevation shift or the transfer of knowledge along the elevation gradient is one very important aspect of it, because climate shift is clear toward the higher elevation. Similarly, bioclimatic gradient is also very well marked here. Second information that we can provide across region is information from one region could be really useful in the another region, or even across the same area. That is very important, as we saw from this climate analog analysis. This is important aspect. So we put two components here: knowledge transfer across the elevation gradient and the knowledge transfer across the region. That is very important for adaptation planning from this developing or reconstructing ecological calendar. That's why it is important here. So I would like to mention here, most of the material used in this presentation are from the part of this ecological calendar project. This consortium have Cornell University from USA, Kunming Institute of Botany, where I am representing right now, University of Beirut, and this Euro-Mediterranean Center on the Climate Change. So lastly, I would like to say this: the ability to anticipate time is a very simple concept, but it's done to establish stability and to create the anticipatory stability. So this research project already graduate several students. Two PhD students are already graduated. One is still working. One postdoc and a master student. So these two are also these PhD students. And this is me. Here is my email address and my detailed published work you can find in Google Scholar and ResearchGate. So thank you for listening. If you have any question, please let me know. You can email me. Thank you, Kishore. Up to you now.
Okay, thank you, Silas. Okay, since we are already running shortage of time and behind the schedule, and our dean is already here for the next session, I would like to put only one question only to you. So that is from Mridul Barman, that in Himalayan rhododendron, in Himalaya, rhododendron height is different in different altitude. What is the biological change there except these heights?
It is a climatic stress, I should say, because I collected these tree rhododendron from around 2000 meter to almost like 4600 meter, and consistently the girth is becoming smaller. But when we cut a section for the dendrochronological work, we found like age difference is not so remarkable. In the higher elevation, more older trees are there compared to the lower elevation. The two region might be high precipitation in the low region and the warmer. So these two factors really enhance vigorous growth of the tree rhododendron in the lower elevation, and these climatic stress make these tree rhododendron really thin and small in the higher elevation. So that is the reason.
Okay, thank you very much. Okay, thanks for your. I personally thank you for your lucid talk, and on behalf of the organizing committee of the Colloquium from Dinhata College and also from the Cooch Panchanan Barma University, I would like to thank Dr. Silas Ranjitka for his elaborate and lucid talk on the ecological calendar, where he has merged so many complex things into a very simple line. So that is your ethnoecology and ecological indicator. And certainly in the days of this climate change, we have many harsh effects. And if you follow his findings, there could be some kind of streamlining with the present scenario. Thank you very much, Silas. And over to you, Joy, sir, for the next session.
Thank you, Dr. Thapa. Am I audible?
Yes,
Dr. Thapa? Yeah. Thank you, sir, for your note of thanks to Dr. Shailesh. Now, I would like to request the Dean of Science, Technical and Vocational Studies, Panchanan Barma University, Dr. Prabir Kumar Haldar, and he is a professor in physics, and he is a member of the advisory committee of this Colloquium. Dr. Haldar, please.
I am audible?
Yes, sir. Perfectly. Okay. Yes, sir. Please deliver your speech, sir.
Thank you. So it is a matter of great pleasure for me to be among the galaxy of students, teachers, several learned faculties. So I am thankful to you all for giving me an opportunity to say something in the valedictory note. So I express my gratitude to our Honorable Vice Chancellor. His profound gesture of love to the faculty and students is truly endeavored. Actually, in these pandemic situations, we are looking for some few questions. Any pandemics look to science for solutions. Where do we stand in this regard? We think the Indian science and technology fraternity has responded well in the COVID-19 situations. And like questions, the Indian science have answers to challenges faced so far and the ability to handle the crisis. And how does India compare to other countries, poorly in some aspects? So we are searching the solution for these questions. And in this juncture, we are just arrange a seminar in this situation, in these areas like our, where the internet connectivity is very low, and the current is not available in 24 hours, and the vicinities of the college and the university are so remote. So such limitation will physically hinder the online system of education, but we should accept these challenges and as an opportunity, and we know the necessity in the mother of inventions. So in this period of crisis, I feel the topics of the Colloquium is quite relevant to the present context for educational institutions. So education is the secondly badly hit sector after the COVID-19 situation. So I understand that you all had a very interesting deliberations in the presence, and I discussions yesterday from a variety of subjects like chemistry, medical history, plant genetic resources. While going through the report, I came to know Dr. Ranjay Shah made us to understand the beginning of origin of life and role of RNA in various steps of life. That was a very interesting lecture. And Dr. is very all know that the tea is one of the important backbone of the economy of the northeastern part of India. So we all know the Darjeeling tea is so famous around the globe. So I Coconut Research Institute, Sri Lanka, who has a vast experience working, has beautifully elaborated the demonstrations of the tea plant across the Asian countries. In today's lecture, so we had a very interesting and relevant derivation of Dr. Jiaul Haq explained strategies to create permanent antibacterial antiviral surface for the prevention of infections either by the introducing cationic polymeric coatings or via interplaying with the surface chemistry. And the second speaker of Dr. Santosh Kumar Shah from Birbal Sani Institute of Paleoscience highlighted another burning from our times and climate change. So, and the lecture will help in the generating widespread awareness about the topic. The last speaker of today, Dr. Silas Ranjitka, introduced us to the concept of the ecological calendars. So I once again thank all the esteemed learned speaker for their deliberations. So lastly, I would like to thank the organizers of the colloquium for organizing such a program, beautiful way. And I would like to express my deep concern about the TIC of uh, uh, Dinhata College. He is
Dinhata College.
suffering a brain stroke. So far I know. So I wish his speedy recovery. So thank you. Thank you.
Thank you, Mr. Halder, for your kind words. I would like to
Hello. Hello.
Yes. Yes.
Hello. Am I audible?
Hello.
Yes, sir. Yes, sir. Perfectly audible, sir.
Hello, Dean sir. Would you please mute your thing?
Yes.
Hello. Okay. Thank you, doctor. Thank you. Hello.
Yes. Yes.
Yes, sir. You are audible.
Acha. Okay. Hello.
Yes, sir. You are audible.
Okay.
Yes,
Okay.
sir.
Okay. Okay. Thank you, Joy sir, for again giving me this opportunity in this session for the discussion and also the winding up the two-day colloquium. So just before making this winding up the colloquium, I see that there are so many participants from around the country and abroad that they are just actively participating in the discussion and also interestingly listening the lectures right from yesterday. And for this, I see there are some some participants. I would like to ask if Dr. Indramani Bhagat is there. Hello. Hello, Dr. Bhagat. Hello, from Nepal. Hello. Okay, if she is not there, I see there. Okay, Dr. Sandeep More. Hello. Hello. Dr. Indramani Bhagat. Hello.
Yes, sir. Yes,
Would you like to have some comment? Would you like
yes,
to have
I'm
some
here.
comment, Dr. Bhagat?
Yes, sir. Yes,
Hello.
I'm here, sir.
Yeah, yeah. Would you like to make a comment on this?
We both are audible.
Sure, sir. Sure.
Okay.
Sure, sir.
Okay, okay. Carry on. Hello.
Okay, sir. Good afternoon, everybody. Honorable Vice Chancellor, Dr. Dev Kumar Mukhopadhyay, Honorable Registrar, Dr. Abdul Qadar Kafili of Cooch, Behar, Panchanan, Barma University. Respected Dean, sir, Dr. Abitan Dutt, distinguished scientist, my colleagues, research scholars. Thank you, sir. You have introduced me. I am Dr. Indramani Bhagat from Nepal. I am associate professor of Cooch graduate campus, Viratnagar. And sir, first of all, I would like to thank the organizing committee, especially Professor Dr. Kishore Thapa, for giving us this opportunity to attend such a wonderful webinar on International Colloquium on Science and Society, Institutional Approaches during Pandemic, 31st July 2020. On behalf of me and my students, all the lectures delivered by distinguished scientists were indeed very interesting and fruitful. And second lecture delivered by Michael Stanley on Chinese medicine gave us a new insight on traditional Chinese method to flourish the health using 5 elements: fire, health, metal, water, and wood. The tea story, origin, domestication, destination, utilization of tea plasm in Asia, delivered by Dr. Muthita Asam, was one of the very fascinating and attention-seizing presentation. As I have done my PhD from North Bengal University on tea pathogen. North Bengal University is the germplasm of tea. It was very interesting to learn about the science behind how different tea species diverge from common ancestor and common region. I also learned that if Chinese Assam and global tea were incorporated, it could enhance the hybrid, vigorous yielding quality and quantity. Dr. G.L. Hawk presented his lecture on engineering antimicrobial surfaces to compact infectious diseases. This is very also interesting. And the fifth lecture delivered by Dr. Santosh Kumar in climate change in Himalayan region and its assessment future perspective. Last lecture delivered by Dr. Silas Ranjitka on ecological calendar in Himalayan and Pamirs. This is a very, he gave the concept of ecological calendar by how this wheat crop are decreases nowadays. And then in last, I would like to congratulate each one of you for making it a good one. I am very pleased to give my feedback and thoughts on this webinar. It was a wonderful experience to me and my students. I hope you all stay safe and healthy during the pandemic. Thank you, sir.
Thank you, Dr. Bhagat, for your comment. If Sandeep More is there, hello.
Yes, am I audible? Am I audible?
Yes.
Okay.
Yes.
Very good afternoon to all of you. Myself, Dr. Sandeep More, Assistant Professor of Botany, Krishnath College, Murshidabad. First of all, thank you, Dr. Thapa, for letting me say a few words in this International Colloquium on Science and Society, Institutional Approaches during Pandemic. First of all, I would like to thank all the esteemed speakers for their wonderful speeches and enlightening us with diverse aspects of science, reasons, and their solutions. So, if I pick one of the talk that is by Dr. Santosh Kumar Shah on climate change in Himalayan region, its assessment and future prospects. So here I would like to say that yes, climate change is a big issue and it is real, but it is not the only one that we face today. Our lives across the planet are being destroyed by global pandemic of this novel coronavirus, So here I have some words to say to all of you that nothing is completely good or bad in the world. Just as there is something bad hidden in every good thing, and in the same way, something good is also hidden in every bad thing. So the same concept applies to the corona pandemic as well. You all know how the corona pandemic has engulfed the whole world today and has had a negative impact on almost every aspect of life. But as I said, in every bad thing, there must be something good hidden. So here I will tell you that this corona pandemic has also some positive impacts in the world. What is the positive impact? If I say most important impact, it is on nature, because pollution level has been decreased, and we are not going out of our house. So we are breathing fresh air. So lastly, I would like to say that when the headlines of rising deaths are coming every day, what we should do, we should look outside the window, we should look at the night sky filled with stars, we should listen to the birds, we should breathe in clean air, and this is the coronavirus silver lining. It's a forced detox of our planet Earth. It has been imposed on us, and we can only hope that we adopt this principle of coexistence, that we come out of this lockdown as more responsible human beings, and we learn to coexistence with other living organisms in the world. And lastly, I would like to request to all of you that do not take our nature and natural resources for granted. I do not take it for granted today in front of you all. Thank you very much. Thank you, Dr. Thapa.
Okay, thank you, Sandeep. I see from South India there is Dr. A.S. Vishwanathan. Amur, are you there? Hello.
Yeah, I'm here, sir. Can you hear me?
Amur, would you like to have some words?
Yeah, at the outset, I would like to thank Professor Kishore Thapa and Dinhata College for organizing such a wonderful international colloquium and for giving me a part.
Introduce yourself.
Yeah, okay. I am A.S. Vishwanathan. I am Assistant Professor in Biosciences, Department of Biosciences at Sri Sathya Sai Institute of Higher Learning, Prashanthi Nilayam in Andhra Pradesh, India. So I take this opportunity to thank especially Professor Kishore Thapa for remembering me from our previous meeting and giving me information about this colloquium in advance so that I could arrange my schedules accordingly and be part of this. I'm very happy to have been part of this two-day international colloquium. Sitting in my desk here, I was able to learn so much, though I would have loved to come there and been part of the thing, but I think the situation is such that I had to remain here. But I was so happy to be part of the entire two-day session. Especially, I would like to make mention of two speakers. All speakers were good. All of them were very informative. But I personally have a lot of interest in ethnobotany. So Dr. Stanley Baker's talk on Chinese medicine was very, very interesting. And it opened my eyes to a lot more about Chinese medicine. I didn't know much about it. I thought it's only about herbs. But I think it's a lot more about so many other factors like emotions, feelings, seasons, and so on. So I think there is a parallel that it draws to our Ayurvedic system of medicine also. Coming to today's talks, I especially enjoyed the last talk by Dr. Silas about the ecological calendar. I happen to teach ecology and conservation, so it gave me so many new insights and a lot more to read about and get to know things about how things happen in different parts of the globe. Thank you very much, sir, for this opportunity to share my feedback also. Thank you very much.
Thank you, Amur. Now, I think we'll have to wind up the program. We have started our two-day session, and also its preparation from long back when we had the first webinar where our Vice Chancellor had said that from here we have a very, very great impact, ill impact of this coronavirus, as we have a very low technical know-how here for imparting the presence in scenes of our education system. We just have only talk and talk system. But yes, he had also mentioned last time that let's take this challenge as an opportunity. And I think we have taken this challenge as an opportunity so that in near future, we are coping up with the other situation or the other alternatives that could be availed for imparting our education. And thanks to our Vice Chancellor, sir, for being with us here last day, or the other day, and also to Dr. Abdul Qadir Safeli, our Honorable Registrar, sir, for giving us his time and also giving a very interesting speech in his as a patron speech that the people here, as we here means in our region surrounded by so many tribals and ethnic communities, they have got so many knowledge about the medical systems and the ethno-medicine system that can well be explored to have our new direction or new sea change in the medical system. And that way, and that way the other day speaker, Dr. Michael Stanley Baker, also mentioned that he has mentioned that how could we make a marriage between the different alternative medicine or the complementary medicine, as what he talked about the Chinese medicine, and as well as with the medicine of Indian system, that is your Ayurveda and Unani, and even the Korean medicine, and also the Shwarikpa from Tibetan medicine. So if we can have a combined flow of this, something great could be explored. And the session that we had today, Dr. Ziaul Haq from California, from School of Medicine, Duke University, and Dr. Santosh Saha from Birbal Sahni Institute of Paleosciences, and Dr. Silas Ranjitka from the Cumin Institute of Botany, China, and also Engine Solution of Natural Innovation from Kathmandu. And we have got so many insights on their expertise. And then with that insight expertise that we have, most of us who are in the teaching profession, and also some of you are in the research profession, they can carry on with these expertise, and that expertise can be implanted on our experiments, on experience later on. And I thank also Dr. Indramani Bhagat and other Amur and Sandeep for giving your comment and feedback, because you see, I could have selected so many persons to have the audio and video feedback, but your feedback from the feedback form will also will give us a boon or inspiration to have our next programs and direction of the program, where should we go. And therefore, I thank all of you, all the participants. You have your active participation. Without your active participation, we would not have made this international colloquium a success. You see, I do consider that this international colloquium a success because of the two things. One, from the cooperation received from all the speakers, and second one, the active participation from all the participants from India and abroad. I also thank Dr. Stanley Baker, my friend from Singapore, and also Ronajai Saha from California, who on our first call and request, he said that, okay, I will be available in that very time, and then he gave us some insight on your his should I say his research finding that has been highlighted to all of us, and that we could get some inception on it. And also to my another friend from Sri Lanka, Kashun, Dr. Kashun. We met several times when he was in China, and then he had an extensive work done on the Asian tea, and as well as he is now doing with his coconut. But during his PhD, he has traveled a lot, and he had come to all the places here and there. And when they had some problem with the Indian tea, and his supervisor had asked me to go there and to be a part of their program, I was there for 2-3 times. And also with his research, and also another person, what he was mentioning, Moses, with their findings, we could get a lot of idea about the tea genetic resources, their domestication, their origin and everything, even the genetic diversity and all. And about today's Silas Ranjitka's lecture. Silas is a very, very hardcore researcher. I also met him in Cumin Institute of Botany, with Kashun and all. And so I'm very much thankful to them, because all of them instantly accepted our request, and within no time we could make this international colloquium possible. And I also express my thanks to the university personals like Professor Pravir Kumar Halda, who is right now here, and he also gave us full support to be the member of our advisory committee. And also I thank our principal, sir, or the TIC, who is now sick and who is now in hospital, and we all are hoping or praying for his very, very quick recovery. Otherwise, he would have been here with us today or yesterday. and also to IQAC cell of Dinhata College, Dr. Devashish Das, and other members of our team. Should I say their labor cannot be explained in words. And also to Subhash sir, who also helped us technically, and he's a faculty of Bengali department of Dinhata College. And it is the success is the combined flow of so many sectors. And therefore, if we think and if we accept this corona effect as the global pandemic, as the challenge, at the same time, then we can take this challenge into an opportunity. And with these words, I wind up this, and I over to Joy sir for the next conduction of the program. Thank you, Joy. Joy sir, hello.
Which would invite the we have got a lot of information in various topics, and we really proud to be part of this international. So, Dr. Kappa?
Yeah. Hello. Yeah. Please put to Manjil.
Hello. Am I audible?
Yes. You are.
On behalf of the organizing committee of the Colloquium, I stand here to express the committee's indebtedness to Dr. Devkumar Mukherjee, Honorable Vice Chancellor, Cooch, Behar, Panchanan, Barma University, for his patronage and his valuable inaugural speech. Our thanks are due to Dr. Abdul Kader Safili, Registrar of the University, for his address and patronage. We are also thankful to Professor Prabir Kumar Haldar, Dean, Faculty of Science, Cooch, Behar, Panchanan, Barma University, for his guidance as a member of the advisory committee and for his time in delivering the valued valedictory lecture. Our special thanks to all our distinguished speakers, namely Dr. Ranajay Saha from USA, Dr. Michael Stanley Baker from Singapore, Dr. M.K. Meghakumbura from Sri Lanka, Dr. Jiaul Haq from USA, Dr. Santosh Kumar Shah from BSIP, Lucknow, and Dr. Silas Ranjitka from Nepal, who have given their time to share their research findings. Our special gratitude to all the participants from India and abroad. Without your participation, our venture would not have been a success. Thank you for taking part in this discussion. Thank you and wish you all good luck and hope to see you all in such an academic gathering next. Have a good day, everyone.
Okay, with the kind words, that is the vote of thanks to all from Manjil sir, I, on behalf of the organizing committee, would declare that this two-day international colloquium is being closed. Thank you all for your participation and for deliberation, and stay blessed, stay safe. Thank you.
I have information for all the participants. Kindly note that you have to fill in both the feedback forms for the generation of certificates, and the certificates will be generated to the registered candidates within 1 week. It will be sent to you via email. Kindly note, you have to fill both the feedback forms. Thank you.